Good morning, and welcome to the Parkinson's Disease Patient and Family Symposium. Thank you so much for joining us for today's webinar. My name is Katie Fagan, and I am the Senior Program Coordinator and a licensed clinical social worker at Northwestern's Parkinson's Disease and Movement Disorders Center in Chicago, Illinois, and I will be moderating today's webinar. Today, in partnership with the Parkinson's Foundation, we are presenting our annual Parkinson's Disease Patient and Family Symposium. A little housekeeping before we get started. After we hear from today's speakers, we will take questions from our web audience during the Q&A panel. Questions can be submitted throughout today's webinar by clicking on the Q&A icon in the black banner on the bottom of your viewing page. We will do our best to answer as many questions as we can. All meeting attendees will be muted through the program. If you are experiencing any technical difficulties, please send us a message using the Q&A icon. We are recording today's virtual symposium, and that recording, along with many of the resources discussed today, including my contact information, will be sent to attendees in an email in the upcoming week. At the end of this program, an evaluation will pop up on your screen. Please fill this out and let us know how we did today. I will now introduce Dr. Danny Bega, who will kick off today's event. Dr. Bega is an associate professor of neurology and the director of the neurology residency program at Northwestern. He has expertise in the care and management of patients with a variety of movement disorders, including Parkinson's disease, and is the director of Northwestern's Huntington's disease and Wilson's disease clinics. His primary area of interest is the study of alternative and non-pharmacologic interventions in movement disorders and their impact on quality of life. And he has received several grants to carry out this research. Welcome, Dr. Bega.
Thank you so much, Katie. I want to just take a second to thank the Parkinson Foundation for all their help in arranging today's event, and also Katie for being our program coordinator and helping coordinate this event, and to all our speakers and panelists who you're going to hear from today. And thank you all for taking some time out of your Saturday to learn about Parkinson's disease. So if you could go to the next slide, please. I'm going to just take a couple minutes here and just fill you in a little bit about our program and our Parkinson's Disease and Movement Disorder Center. I'm coming to you live from downtown Chicago, where our downtown center is located. And our mission is to provide a comprehensive, multidisciplinary care to people with Parkinson's disease and other movement disorders, while also doing research into causes, into potential treatments and for the disease. And so, today we're trying to achieve one of our missions, which is education. We really think about the three pillars of clinical care for patients and families, research, and education, and that's part of why we're here today. Next slide. And can you advance that again, just to complete the slide? Parkinson's disease is a complicated disorder, one that involves more than just movements. And this is a depiction here of showing that, part of what we see in people with Parkinson's disease is visible above the surface. We think about tremors and walking problems, which can be easily seen and which we have ways of treating. We also know that a lot of what people experience with Parkinson's disease is different from person to person. And there's an array of different symptoms that people might experience that are potentially less visible beneath the surface, that can include anything from fatigue to cognitive issues, which you're going to hear about today, to things like dizziness or sleep problems with sense of smell, and other symptoms that can kind of make up the day-to-day life of someone with Parkinson's disease, and really leads to a need for a multidisciplinary approach to treatment that goes beyond actually even the movement disorder specialist. If you can go to the next slide. And so we really believe that it's important for someone with Parkinson's disease to have a comprehensive care model, which involves a neurologist with expertise in Parkinson's disease, along with other specialists, nurses, rehabilitation services like the ones we have at our affiliate, the Shirley Ryan Ability Lab. So physiatrists, PTs, OTs, speech therapists, exercise physiologists, social workers, nutritionists, surgical team members as well, and research team members that help us with clinical trials. And so part of what makes a Parkinson's Center of Excellence is this sort of combination of different specialists who can approach the disease and work together to provide a more comprehensive and holistic approach to care. Next slide. With that, we offer a lot of that in our downtown campus, but I also want to highlight we have a lot of specialized programs, and we also offer expert care at other sites around Illinois. So first, at our downtown site, in addition to our standard movement disorders clinic, we also have specialized programs. We have a specialized clinic in palliative care in movement disorders. We have a specialized clinic for cognitive problems, for genetics. We have a specialized clinic for atypical Parkinsonism. This would include things like PSP or MSA, some of the rarer forms of Parkinsonism. We have a Spanish-speaking movement disorders clinic. We have an active surgical program and a lot of research going on. We also are offering a lot of specialized care at some of our other sites, including many of you today here who probably get your care at Lake Forest or Central DuPage. We also have a team at Palos and in Glenview. So we really have expanded well beyond our downtown campus. Next slide. The Parkinson Foundation, who's partnering with us for this event today, has designated us a Center of Excellence. This is a badge of honor that we have, that we share at our downtown site, at our Lake Forest site, and at our Central DuPage site as well. And this means that we offer comprehensive care with different team members that I mentioned, along with research and education and outreach into the community. Next slide. So this is just showing you our downtown site. We have a lot of movement disorder specialists ready and eager to see people with Parkinson's disease and as part of this multidisciplinary team. And if you go to the next slide, we often forget to mention we have a really extensive research team doing, the behind the scenes work. These are people that aren't seeing patients on a day-to-day basis, but they're in the lab. They're doing a lot of the important work that eventually gets to the patients in terms of the science that leads to really important developments in the field. And this is also, these scientists are also at our downtown site. Next slide. Other team members, we have trainees learning to be specialists in movement disorders, so fellows. We have residents and medical students. We have nurses and social workers, mental health counselors, rehab team. We have neuropsychiatrists and neuropsychologists working with us. We have a genetic counselor and then a neurosurgical team. Next slide. And then hopefully, I hope many of you are aware of all the programs that we have that are all free and that we offer in the community. This was scrolling through ahead of the talk, and we'll be scrolling through again with some of these opportunities during the break. But we have support groups that are both, we have some online ones and some in-person ones. We have educational events throughout the year. So while this is our biggest annual symposium, we do smaller educational events throughout the year. We're also introducing some Spanish education events as well. We have exercise programs that are virtual. We have in-person programs for dance and improv therapy. So a lot of good things that you can take advantage of. Katie Fagan, our coordinator, helps set up a lot and organize a lot of these programs. Next slide. And we're really leaders in translational research. And what that means is really bringing together the scientists who do the bench work and the clinic and the patient care that we do at the bedside, and trying to span that with the research we do. Next slide. And so there's a lot that you're going to learn about today as you hear from the head of our division, Dr. Tanya Simuni, who's going to talk about research. You'll hear about the importance of genetics and other factors in what's happening in the field of Parkinson's disease. Next slide. You'll also maybe hear about some of the big headlines that have come up in the last year. I know a lot of my patients have asked me about this, about new biomarkers, which you're going to hear about from Dr. Rizwan Akhtar. And then some of the way we're staging Parkinson's today. Also, Dr. Simuni has been one of the leaders in this area. Next slide. And in addition to the research, there's also new therapies that are available now at the bedside to help with symptoms. And this has taken the form of surgical therapies or new medications. And there are some that we've been waiting on for a few years, like infusions that we're hoping will be available soon to treat symptoms in Parkinson's disease. Next slide. We really encourage everyone to be active and exercise. That's why we have so many different community programs to work on exercise and mind-body interventions to improve quality of life. Next slide. And many of you are already participating in this. These are all pictures that we've gotten from all of you of your participation and activities that really can make a huge impact on your day-to-day life with Parkinson's disease. Next slide. So with that, I'm gonna hand it over in a moment to Jessica Barsh from the Parkinson Foundation. I just want to put a few reminders out there. Be optimistic about the horizon. Again, you're going to hear about research updates from Dr. Simuni today. I really recommend seeking expert opinion at specialized Parkinson centers for the reasons I showed you, that multidisciplinary care approach, for exposure to clinical trials, which we're happy to talk to you about, and for educational resources and support groups. And then, we have potent medications today to help treat Parkinson's disease symptoms. Luckily, we have new ones coming out all the time. And then remember to be active. That's really important. It can really make a difference in your day-to-day life. With that, I'm going to thank you for your attention and open up the next speaker at our symposium, which is Jessica Barch from the Parkinson Foundation. Oh, I also real briefly want to also thank our sponsors. Thanks to the Parkinson Foundation, of course, our partner in this event, but also Amneal, AbbVie, and Supernus for their support in this event. Thank you.
Hi, everyone. Thank you so much for the introduction, Dr. Bega. My name is Jessica Barch. I am the Senior Manager of Community Education and Outreach for the Parkinson's Foundation Midwest Chapter. And the mission of the Parkinson's Foundation is essentially to make the lives better of anyone affected by this disease. Whether you are living with Parkinson's, caring for someone with Parkinson's, or working to end this disease, we are here to support you. Next. To support our global community, we focus on 3 goals: improving care for people living with PD through centers of excellence like Northwestern University Movement Disorder Center, advancing research toward a cure, and empowering the community with educational resources. Next. The Parkinson's Foundation has a wide array of free resources. We have educational books, a podcast, our Aware and Care hospitalization kit, a helpline staffed by experts, our website parkinson.org, and more. Next, please. And Moving Day Chicago is right around the corner. Come join us on Sunday, October 29th to honor loved ones with Parkinson's disease, raise awareness, and to celebrate movement, which is proven to help manage Parkinson's symptoms. To learn more, visit movingdaychicago.org. Next. Did you know the Parkinson's Foundation is offering genetic testing at no cost for people with a PD diagnosis? This initiative is called PD Generation, and participation can either be done in person at one of our participating centers of excellence sites, such as Northwestern, or from your own home through a telemedicine appointment and an at-home cheek swab kit. To learn more, visit parkinson.org/PDGeneration. Next. Are you caring for someone with Parkinson's disease? Our Care Partners Summit brings care partners together to provide resources to make life better for themselves and their loved one with Parkinson's disease. This year's summit will focus on how to communicate through difficult topics and find supportive connections. Join us online at home via Zoom on Saturday, November 11th. To register or learn more, visit the link parkinson.org/summitweb. Next, please. And lastly, I wanted to inform you of our virtual series, which is a weekly series called PD Health at Home. This series is focused on Mondays, Wednesdays, and Fridays and we look at mindfulness, wellness, and fitness. To learn more or to register, please visit parkinson.org/pdhealth. Thank you, everyone, for your time today, and I will pass it now to Katie.
Thank you so much, Jessica. And thank you to Dr. Bega for all of that information that was provided in our introduction. Before we begin today's presentations, I want to remind you all of our agenda. So we obviously are finishing up our welcoming and opening remarks. Our upcoming presentation is Synuclein Biomarkers by Dr. Rizwan Akhtar. At 10:55, we will have our therapeutic pipeline in early Parkinson's disease and genetics presented by Dr. Tanya Simuni. And then we will follow that up with a 10-minute break. At the conclusion of that break, Dr. Michael Mercury will talk to us about dementia and Parkinson's disease. And then for our last half hour of our presentation, our webinar today, we will have our clinician Q&A panel, and then we will wrap everything up at 1 o'clock. Thank you all so much again for being here today. So now it is my pleasure to welcome Dr. Rizwan Akhtar. Dr. Akhtar is a physician scientist in the Division of Movement Disorders at Northwestern University's Feinberg School of Medicine. His research laboratory investigates diagnostic and prognostic biomarkers for Parkinson's disease and related neurodegenerative disorders. He oversees several clinical trials for PD at Northwestern and is the principal investigator for the Movement Disorders Biorepository at Northwestern. Dr. Akhtar frequently gives lectures to national and international audiences, and he sees patients in the Regenstein Center for Neurological Care. I hope you enjoy his presentation.
Okay, hello everybody. Welcome to today's talk. Today I'll be talking about alpha-synuclein biomarkers in Parkinson's disease. And my name is Rizwan Akhtar, and I'm at Northwestern University. Here are my disclosures for today's talk. There's nothing particularly relevant for what I'll be talking about today. So before I talk about alpha-synuclein and biomarkers, I thought I would give a little bit of perspective about Parkinson's disease and what happens in the brain when this disease begins. And the primary problem in Parkinson's disease has to do with accumulation of proteins in the brain in structures called Lewy bodies, and that's shown here in this picture. These were appreciated in the early 1900s, and that was about 100 years after the diagnosis of Parkinson's disease was first described clinically. So it took about 100 years for scientists to realize that this process is happening in the brains of patients with Parkinson's disease. And over time, it became accepted that to have Parkinson's disease, you had to have these inclusions or aggregates in the brain. And now we know that these initial aggregates that were described many, many years ago now are not alone. And there's a number of different aggregations that happen in the brain of patients with Parkinson's disease as the disease is beginning. But what was not clear until really the late 1990s was what was the proteins involved in these aggregations. And that's where alpha-synuclein comes into the picture. So this was first identified in 1997 by looking at patients and their family members who had a very strong family history and risk of getting Parkinson's disease. And by carefully looking at their genetics, researchers were able to determine that this family carried a variant or a mutation in a protein called alpha-synuclein. Alpha-synuclein is a small protein. This is a cartoon of what it looks like. And it is a normal protein that's expressed in the brain, and specifically in cells in the brain called neurons. And we're still understanding exactly what synuclein does in the normal state, but we know that you need alpha-synuclein in order to have a well-functioning brain. The problem in Parkinson's disease appears that instead of just being normal, the alpha-synuclein protein begins to misbehave. And that process of the protein starting to misbehave is critical for the understanding of why Parkinson's disease happens. So there are a small number of proteins, and the list is growing in the brain, that can lead to these types of aggregations. And alpha-synuclein is only one of those proteins. But all of these proteins share certain features. And one of those features is that these proteins are able to misfold and aggregate. And what that means is that all proteins that you express in yourself, in your body, are all folded in a certain way. And by doing that, they adopt a structure that helps them do their function. And that's shown in this cartoon right here. And through no fault of their own, usually these proteins can sometimes misfold. And the cell has a variety of different mechanisms to try to refold those proteins to get them back in the way they're supposed to be. So as these proteins transition from a normal folded state to a misfolded state, the cell is trying to do things to try to get it to refold and go back to its normal job. Now, sometimes these misfolded proteins cannot be refolded, and cells have ways of dealing with those misfolded proteins. They might break them down into small parts and recycle them, or they might send them on to the trash can of the cell to get rid of them completely. Now, the problem is that as these proteins like alpha-synuclein are misfolding, sometimes they can start to aggregate. And what that means is that these misfolded proteins begin to stick together and create longer structures that are progressively more difficult for the cell to handle. These are shown here. The misfolded proteins begin to aggregate first as sheets, and then finally these sheets start stacking on one another and create these very large, sticky aggregations in the cell that the cell can't really do much with. It isn't able to recycle it, isn't able to break it down and send it to the trash can. And so these aggregations of this protein are what we think are present inside of those Lewy bodies that I showed earlier. In the case of Lewy bodies, they're filled with alpha-synuclein. But we've now learned that there are a number of other proteins that also can go through this process, that can transition from being normally folded to misfolded and aggregated. And when that happens, these proteins deposit in a variety of different structures that we see in the brain and we know are associated with different diseases. So for example, in Alzheimer's disease, the brain begins to fill with something called amyloid beta plaques. These are misfolded, these are aggregations containing a protein called amyloid beta. Alzheimer's disease also has aggregations containing the protein tau, and other neurodegenerative diseases also share the same feature, that there are these proteins that are misfolding, aggregating, and then those aggregates lead to problems in the cell. And so we know now that amyloidogenic proteins are central to all neurodegenerative diseases. And amyloidogenic is the word that means able to create protein amyloids, which is what we call these more complex structures of misfolded proteins. So if we could understand this process and figure out how to prevent these proteins from transitioning from a normal state to an abnormal state, we could make major impact in a number of different diseases, not just Parkinson's disease. So at some point, researchers asked, when does this process first start and how does it change over time? And we've learned a little bit about this when looking at autopsy series from patients with Parkinson's disease. These are series where we take patients who have donated their brains to science and we look closely at their brains to try to understand what happened while those patients were living. And in doing that, it appears that this process of protein aggregation and misfolding does not occur randomly in the brain. Instead, it seems to follow a pattern. It seems to start in regions of the brain called the brainstem, and maybe even before the brainstem, even in other parts of the body, not the brain. But once this process starts happening in the brainstem, we think that some early symptoms of Parkinson's disease may emerge. And then as time goes on, this process seems to spread. So these proteins that are misfolding now involve the midbrain. And then from the midbrain, it involves other parts of the brain, like the hippocampus and parts of the cerebral cortex. Now, this is a hypothesis in patients because we cannot follow this process in one person. We have to look at a whole group of people and compare where we see these abnormalities in order to put together this kind of model. But there's a reasonable amount of data now that suggests, mostly in the lab, that suggests that this process does happen, that there is a initial misfolding event that affects Parkinson, that affects alpha-synuclein. And then as time goes on, that process begins to affect other regions of the brain that are connected by synapses. Now, we don't have a biomarker as yet for this process happening in the brain in a living person. And this is very important to have. And we'll come back to this a little bit later in terms of what biomarkers we do have now that can maybe help us understand more about this process. So I mentioned that there's a number of studies now in the lab that suggest that this process is happening, and I'll go over a little bit of that here. So we can take protein like alpha-synuclein and mistreat it in the lab in a test tube to create these misfolded structures that I showed earlier. And we think that these misfolded structures, these fibrils of protein, can self-propagate as well, and I'll talk about that a little bit later. So scientists have been able to take this material, again, from the test tube and inject it into mice brain to see what happens. And when you do that, what you find is that the aggregations of alpha-synuclein first start in the region where this material was injected, but then over time tends to spread to other parts of the brain in a mouse. And this data suggests that our hypothesis might be true for patients as well, that in patients, the misfolded protein may begin in one part of the brain and spread over time. And this data now has been replicated a number of times in different animals, not just mice. And it all suggests that if you take this protein in the lab, create this misfolded version of it, you can take that protein, inject it into an animal, and watch it spread through the brain. So how does that process work? Well, we don't really know how these misfolded proteins spread to the brain. There's a variety of different hypotheses. One is that perhaps one cell actually packages that misfolded protein, and the package transmits from one cell to the adjacent cell, and that's how the process moves forward. But there's a variety of other hypotheses, and we're working on this very closely to try to figure out how this process happens, and then potentially how to stop it. So that's an introduction to alpha-synuclein. I'm going to shift gears now and talk about biomarkers. So what is a biomarker? I've written here a definition of a biomarker, and I'll read it. The definition is a distinct and measurable characteristic that is an indicator of an underlying biological process. So it's something that you can measure in a patient that tells you something about their underlying biology. So a good example of a biomarker is the annual test for high cholesterol that many primary care providers do for us every year. We go in, we have blood drawn, and our total cholesterol and other lipids are measured. What does that tell us? That tells us that if your cholesterol is elevated, you might be at higher risk of having a heart attack or a stroke. And that makes a clinical impact in some people. If it's abnormal, your doctor might tell you, you need to exercise more, we need to talk about diet, maybe you need to start a medication to reduce your cholesterol. So the cholesterol is the biomarker. It's measured in the blood, and it tells you something about the underlying risk of a person having a cardiovascular event. So in the case of Parkinson's disease, what is the biomarker that we would like to see? Well, one would be something that would indicate the loss of cells in the brain, or maybe even the spread or the misfolding of alpha-synuclein that's happening. And we already have some biomarkers in Parkinson's disease that we do use clinically. And so that's described here. In Parkinson's disease, one of the major pathologic hallmarks of the disease is a loss of the dopamine expressing cells in a region of the brain called the substantia nigra. So these pictures here are taken from human brain. This is a zoomed in picture under the microscope. And in a healthy subject, you can see that these cells contain, there are many cells here that are dopaminergic, and they contain dopamine, and they're doing their normal function by sending that dopamine to another region of the brain here called the striatum. And we see that in this region of the brain, there's a number of different transporters that are expressed that help move dopamine around. So that's what we see in a healthy subject. In a person with Parkinson's disease, you see that many of those cells are missing, and the resulting connections of those cells in the striatum are no longer there. The cells are lost in the nigra, and the connections are no longer there in the striatum. So this is the underlying biologic process that we might be interested in Parkinson's disease. If we can measure this, it would be useful. And it turns out that for this particular process, we do have a biomarker. That biomarker is shown in the middle here. This is a SPECT study, which is a type of radiology study, and it uses a tracer called DAT scan in this case. And this tracer is a chemical that travels to the brain and binds to transporters that are expressed at the cells that we are seeing here that are lost. And so when that tracer binds to that region, a patient can lay down in a scanner, have a picture taken of that tracer. And by doing so, you can see that there is a decrease in the amount of the tracer binding in the striatum in a patient with Parkinson's disease as compared to the healthy subject. So again, these pictures here are taken from a patient who's died, right? We're looking at this person's brain after they've passed away. These pictures are taken from a living patient. So in this case, the biomarker is the dopamine transporter ligand. It's binding to the dopamine transporters here, and it's indicating something about an underlying biologic process. In addition to that, we can quantify the loss of the dopamine transporters here and make more quantitative assessments of the change of this biomarker. So we already have a good biomarker for dopamine transporter expression in the brain. What we are lacking is a good biomarker for alpha-synuclein, and I'll come to that in a moment. So what are biomarkers? How could we use them in neurodegenerative disease in general? Well, they're very important for neurodegenerative disease because we think that these biomarkers start to change early on in the disease course. And this is now an old picture published in 2013, but modifications of this concept have been published many times, and we discussed this extensively in neurodegenerative disease. And what this is showing is the idea that biomarkers can start to become positive or become abnormal relatively early in the course of someone's disease. So this particular figure describes what we think might happen in a disease like Alzheimer's disease. So as time goes on, time is on the x-axis here, as time goes on, patients with Alzheimer's disease or maybe mild cognitive impairment might start developing cognitive symptoms. Those are shown here in green. And at some point, those cognitive symptoms might become severe enough that the patient presents to see a doctor, the doctor evaluates them, and may diagnose them with something like mild cognitive impairment or dementia. And that happens at a particular time in the patient's life. However, at that particular time, we think that there are already a number of biomarkers that have changed in that patient. Those are shown here by these particular lines. So for example, changes on MRI might happen well before a patient presents with symptoms and is evaluated. Sometimes there can be changes in cerebrospinal fluid biomarkers or other types of PET ligands that can all become abnormal early on. So understanding this process, the processes that happen in this phase of an illness are really important. If we want to prevent someone from developing enough symptoms to merit a diagnosis of mild cognitive impairment or dementia that happens later on, we need to understand these biomarker changes early on. And if we can identify patients who are at risk of developing the syndrome later on, maybe we can make an intervention that will make a meaningful impact in that patient. So I hope I've convinced you we need a biomarker for alpha-synuclein aggregation in Parkinson's disease. We already have a reasonable biomarker for dopamine expression. So what could it do in PD? Well, the list is pretty long, But one very important concept is that it would enable us to make a diagnosis earlier, and potentially a diagnosis that's based on a person's biology, not symptomatology. So I'll go back a moment and show this slide again. If we diagnosed a patient in this example with mild cognitive impairment or Alzheimer's disease in this phase of their illness, they don't have symptoms yet. Remember, the symptoms are in green. But their diagnosis is based on the biology that is changing. So even though they don't have symptoms, we think that maybe some of the biology that's leading to Alzheimer's disease is present, and as such, we might make a diagnosis like that in that phase. And so that's something that could happen with Parkinson's too, that if we could measure alpha-synuclein misfolding or aggregation in the brain, even though a patient may not have symptoms of Parkinson's disease yet, we might be able to diagnose them with that disease if we base the diagnosis on biology rather than symptomatology. So that's a very important potential benefit of having a biomarker. In addition to that, it'll help us make a more accurate diagnosis. We know that some patients, when they first present with Parkinson's disease, are a little bit confusing to their doctors. And over this next year or two, they may actually present with a syndrome other than Parkinson's disease. Even though the first visit suggested that they might have Parkinson's disease, by the second or third visit, it might be clear that they may have something else. So if we had a biomarker for alpha-synuclein, we might be able to make a more accurate diagnosis in those early time points. It's very important to try to have a biomarker to tell us whether or not an intervention that we are testing in a clinical trial is working. And this is something that a biomarker for alpha-synuclein would be of great help to do. We would be able to see if there are changes in alpha-synuclein misfolding or other processes happening with alpha-synuclein in response to a therapy we're testing in a clinical trial. And this is the concept of target engagement. So it's another important area where a biomarker could be helpful. And then lastly, clinical trials would benefit if a biomarker for alpha-synuclein was available, because we would be able to ensure that the patients that we recruit in a clinical trial actually have the disease that we're trying to study. And despite all of our efforts, even at tertiary care centers, the diagnostic accuracy of Parkinson's disease is not 100%, it's less. And so something like a biomarker for alpha-synuclein might help us exclude those patients that don't actually have that disease, so we don't accidentally enroll them in a trial for PD. So I'm going to talk now about alpha-synuclein seeding assays. And this is the most recent biomarker that has been developed for alpha-synuclein that is engendering a lot of excitement in our field. And I'd like to discuss what this means, and then we'll talk about some of the data in a few minutes. So I mentioned earlier that in the test tube, in the lab, you can take monomers or alpha-synuclein proteins that are folded normally and mistreat them so that they begin to misfold, aggregate, and eventually create these long fibrils. And we've understood this process for a long time. What we have also realized is that if you take the final product of that misfolding process, break it apart, maybe mistreat it even more, it itself, those pieces can serve as a seed to restart this process from the beginning. And this is the concept of self-propagation with respect to fibrillization of proteins like alpha-synuclein. So you could take alpha-synuclein, mistreat it initially, it starts to misfold. We then can break apart those aggregates, again in the test tube, and use those seeds to generate new preparations of the misfolded protein. So this concept was adopted and adapted when alpha-synuclein seeding assays were generated. And so what is the assay? How does the assay really work fundamentally? Well, the first step is to take a seed of alpha-synuclein and start the process of aggregation. This seed, in the context of biomarker, would come from the patient's sample themselves. So that seed starts the process of aggregation. Over time, that aggregation becomes self-propagating, and we have certain chemical dyes that can show us when these aggregates are occurring, and it takes some time, but we have fluorometers and other devices that can measure the aggregation of these proteins over time. And so that's shown here in this green line, where as time goes on, if seeds were present in the sample to begin with, we will start to see fluorescent tags or fluorescence that is emerging from these misfolded structures that are occurring. And that can be measured in this machine. And once a certain level of misfolded proteins are present, we would say that this assay is positive for alpha-synuclein seeding. So if the seed was not present, let's say a patient who doesn't have Parkinson's submits a sample for this type of assay, or a healthy subject, the process of aggregation never starts. And so as time goes on, that sample never generates any aggregates. There's no fluorescence to be measured. And by the end of the assay, that sample remains negative. So this is how you can distinguish samples that had a seed at the beginning for the aggregation from those that didn't. So the names of these assays there's a couple of different names in the literature, but the main one that we'll be talking about today is called real-time quaking-induced conversion, or RT-QuIC. Another assay is protein misfolding cyclic amplification assay, or PMCA. They both work in similar ways. The main difference between these two assays is the way that the pieces are made from these fibrils. In the case of the RT-QuIC, the proteins are shaken, and that shaking causes them to break apart. Whereas in the PMCA assay, sonication or ultra high frequency sound waves are used to break apart these fibrils. But in both situations, the fibrils are broken apart, allowed to undergo this self-propagating process. So the first few papers in alpha-synuclein seeding in the context of Parkinson's disease were published several years ago now. And this was one of the earliest papers that looked at alpha-synuclein RT-QuIC in the spinal fluid of patients. And what you could see is that if you compared control subjects, who are shown here in red, as you allow the assay to progress over time, these normal subjects did not have any seeds in their spinal fluid to promote the aggregation that we see on the fluorescence. In comparison to that, if you look at a patient who has, in this case, dementia with Lewy bodies, which is a synucleinopathy, you can see that after about 50 hours in this machine, the seed has led to enough aggregates that we start to see this fluorescence signal. And so if you look at this data, you will very clearly see that there is a clear difference between those patients who have synuclein seeds, which is shown here in blue, compared to the healthy subjects who do not have any seeds. And this is done again here in this. These are 4 control subjects and 4 patients with Parkinson's disease. This is all spinal fluid in this particular picture. And what you can see is that those patients with PD, after about, in this case, almost 100 hours, start to show this fluorescence that emerges, indicating that there's misfolded aggregates happening in the reaction in the machine, whereas healthy subjects don't have. So this was one of the earlier papers that have come out. And this year, a paper was published in a journal called Nature Protocols, which described how to do these assays in the lab and really demonstrated over the course of several years optimization of this assay so that it works very well to distinguish subjects with Parkinson's disease from healthy subjects. And so here's this a depiction of what happens. Again, healthy subjects don't have any seeds in their sample, so they're not able to generate any of these aggregates, and there's no fluorescence that's detected over time. Patients with Parkinson's disease, it takes 100+ hours for this machine to keep reading, but eventually enough fluorescence emerges that we can measure that, and it continues to go up until all of the monomers or the substrate in the reaction mix are used up. And we also now know that other synucleinopathies, like multiple system atrophy, also have abnormal alpha-synuclein seeding, except in other diseases, the kinetics of that seeding differ. And they differ sufficiently that we think that maybe we can distinguish a patient with Parkinson's disease from a patient with multiple system atrophy on the basis of the kinetics on this assay. Obviously, we have to confirm that and repeat these, but it does appear that not all alpha-synuclein diseases act the same way in these assays, and we can potentially use that difference to develop additional diagnostic biomarkers. So again, this is a paper that was published, I think, in April of this year, and described how to do these assays in the lab. Soon thereafter, another paper was published. This is in Lancet Neurology, from the Parkinson's Progression Marker Initiative, that looked at almost over 1,100 participants in that study to ask whether or not the spinal fluid that were collected as a part of that study contained seeds that could be detected with this assay. And if so, did those seeds differ in patients with Parkinson's disease compared to other patients that were recruited as a part of this study? So I mentioned this is 1,100 or so patients, over 30 centers across the globe, and this took 9 years to collect all of these samples. But in this paper, it was very clear, and I'm blowing up the data that was shown here in this paper to really highlight what was found. And that is that if you looked at Parkinson's disease subjects in this study, 88% of the spinal fluids in patients with Parkinson's disease had positive alpha-synuclein seeding. And in comparison, the healthy subjects who were in PPMI in this study had very rare positivity on alpha-synuclein. You can see that this distinction here is substantial. And it suggests that this seeding assay, as was done in this project, would be very useful in making a diagnosis of Parkinson's disease, because it would identify those who had the disease and not identify healthy subjects. Now, obviously, it's not perfect, right? There were some patients with Parkinson's disease, about 12% of them in this study, who did not have a positive alpha-synuclein seeding. And again, there were 6 subjects who we thought were healthy in PPMI who happened to have a positive alpha-synuclein seeding assay. So what does that tell us? Well, it tells us that maybe there are some patients with PD who don't have alpha-synuclein. Maybe they have some other protein, or maybe something completely different. They look like Parkinson's disease, but they don't seem to have alpha-synuclein seeding. And we do know that there are some patients who carry certain gene variants that do appear to not have the traditional Lewy bodies when they go to autopsy. What about these healthy subjects? What about these 6 that had positive synuclein seeding? Well, I mentioned earlier that these biomarkers often change before symptoms emerge. So these 6 individuals, perhaps we're examining them too early. And if we wait another 5 years or 10 years or 20 years, maybe they'll have symptoms of Parkinson's disease then. And what we're seeing here is an abnormality on the biomarker prior to the onset of clinical symptoms. Now, I don't know who those 6 people are. I don't know if they're healthy or not. Maybe they happen to have a positive synuclein seeding by chance, but we think that that probably doesn't happen that often. So here's some more data from this paper. This is data that shows all the patients with sporadic Parkinson's disease. The blue dots are the ones who have a positive synuclein seeding on their CSF. The open circles are those who are negative. The x-axis here shows the performance on something called the UPSIT, which is the University of Pennsylvania Smell Identification Test. It is a test to see how well you can smell, and patients who who have lost their sense of smell tend to perform poorly on this test, and so their scores will be all down here towards the bottom. And on the y-axis here is a quantitative measure of participants' dopamine transporter expression. So I mentioned way back that we can measure dopamine transporters using a SPECT scan. And so people who are towards the bottom of the y-axis have relatively less dopamine transporter in their brain compared to those that are higher up. And the 2 lines that are shown here are norms to sort of separate those we think are abnormal in the case of both biomarkers in this quadrant from those that are normal, who are over here or over here. So participants over here have a good sense of smell, but maybe have lost some dopamine expression. And people who are in this quadrant here have bad sense of smell. They can't smell very well, but they tend to have all their dopamine transporters. And then there's patients up here who are normal on both. And because this data is from patients who have sporadic Parkinson's disease, there's not very many patients in this group. So what you can glean from this is a few things. The first is that having a positive synuclein seeding assay tends to associate very strongly in the patients who have smell loss. But the patients who happen to have lost their sense of smell also are very likely to have an abnormal synuclein seeding, and more so than the patients who have lost their dopamine transporter expression. And I say that because there's a reasonable number of people who are out here who have open circles who have lost their dopamine transporters, but they don't have a positive synuclein seeding. So this suggests that hyposmia is a very strong, or loss of sense of smell, hyposmia, is this very strong associator of positive synuclein seeding in sporadic PD subjects. So this paper also looked at a variety of different clinical features, try to determine, were there clinical measures that associated with positive versus abnormal alpha-synuclein. And the long story short is that in this particular paper, there were only modest associations between different clinical features and whether or not a participant had a positive versus a negative synuclein seeding assay. That was a little bit not true in the LRRK2 patients. These are patients who carry a variant in LRRK2, which is a gene that increases, that if you have a variant, may increase your risk of getting Parkinson's disease. In that group, there were some clinical associations, in particular, women who had LRRK2 variants tended to have a lower likelihood than men of having a positive synuclein seeding assay. That was not seen in the other groups. In the other groups, it was about equal between men and women. But overall, a lot of the clinical features that were measured in this paper didn't show a strong association with the positive versus the negative synuclein seeding. So what does that mean? That means that we still have a lot to learn, because we do think that the presence of these seeds and the presence of alpha-synuclein aggregation is important for the diagnosis of Parkinson's disease. In other words, to distinguish whether you do have the disease or you don't have the disease, this biomarker will be very helpful. We still have to figure out how to use this biomarker to tell us about how severe a patient's Parkinson's disease. If you believe the hypothesis that I showed earlier, that the alpha-synuclein tends to spread over time, it would make sense that those people who had more alpha-synuclein aggregates that we can measure should have more severe clinical disease. And those people who have less alpha-synuclein aggregation should have less clinically severe disease. And so we still need to test that to determine if that's true or not. At least this data doesn't suggest that that's the case, but we need to replicate this and look at a larger number of people. Okay, so what's the future of these assays and for alpha-synuclein biomarkers in general? Well, I had mentioned this earlier, but I want to come back to this again. One of the things that we hope we could potentially do with a biomarker for alpha-synuclein seeding is to create the concept of a biologic definition of the disease. So in 1817, James Parkinson described the syndrome that we still look for today in the office. We look for resting tremor, we look for stiffness, we look for slowness of movement, and the presence of these symptoms in the right context can help us make a diagnosis of Parkinson's disease. But we want to take that diagnosis from the clinical realm and move it into the biologic realm. We want to be able to have a biologic measure, a biomarker that will help us make the diagnosis. And I think abnormal alpha-synuclein seeding might be that biomarker. I also mentioned that fine-tuning of groups in clinical trials is important. So as we do it now, we recruit a large number of patients to trials. They're all very heterogeneous. No patient is exactly like the other. And so because of that heterogeneity, it can sometimes be hard to identify the interventions that make a difference. And we may be missing some because the groups are too dissimilar. So if we used a alpha-synuclein seeding assay as a biomarker in enrichment of clinical trial cohorts, we might be able to fine-tune the groups and make sure that groups that are being compared against one another are more homogeneous. So what do we need to do in the future? Well, we need to confirm these results. As I mentioned, all of the data I showed today was published this year. So additional labs need to do to confirm these results. And the way science works is that we like to replicate things more than once to know that it's true. The assays, as of right now, all rely on cerebrospinal fluid, which you have to collect with a lumbar puncture. And that's a little bit more challenging for patients as well as for doctors to do. So we would very much like to transition this assay to use blood or skin. And then a big question that we don't know yet is, does the degree of seeds that we measure in patients change over time? Does it go up? Does it stay the same? Does it go down? Does it go down in the response to certain interventions that we're making? This is very important to determine, and I'm hoping that we'll be able to do that. And then lastly, I'll just mention again that this idea of a biologic staging system for Parkinson's disease is being discussed in our field quite aggressively. This is a paper from colleagues of mine that was published that kind of highlighted a proposal for how to create this biologic staging system. And so stay tuned, because we are working on this actively, and hopefully then in the short term, this will be a part of clinical care. So with that, I'll close, and I thank all of you for your attention.
Thank you so much to Dr. Akhtar. That was an excellent presentation. Now we will hear from Dr. Tanya Simuni. Dr. Simuni heads the Northwestern University Feinberg School of Medicine, Parkinson's Disease and Movement Disorder Center, which is recognized by the Parkinson's Foundation as a center of excellence. She is a world-renowned expert in Parkinson's disease modification studies and has more than 100 publications in peer-reviewed scientific journals and book chapters, and has lectured nationally and internationally on Parkinson's disease and other movement disorders.
Good morning. I would like to join Katie and Dr. Bega in welcoming you to Northwestern Medicine 22nd Annual Patient and Family Parkinson's Symposium. I am honored to be on the podium. And before I start, I really want to give huge thank you to the organizers for putting tremendous amount of work in preparing this symposium. We spent a lot of time discussing whether it should be virtual or in person. We decided on the virtual framework, as you can see, for the reason that we are reaching out to hundreds of people, and also the opportunity for you to return back to the slides and to the presentations and share it with the family members if interested so. So without further ado, the topic that I will speak on is Parkinson's disease therapeutic pipeline, October 7, 2023. And as I traditionally do, I usually cover with very wide brush strokes the questions or topics that are new since the last year that patients ask in the clinic and that you've seen in the recent publications. So without further ado, these are my disclosures, and some of these programs I will be speaking in the course of my presentation. And these are the talk objectives. I will start with new symptomatic therapies and the pipeline. I will briefly cover current state of treatment of early Parkinson's disease. And the focus will be on experimental therapies aimed to slow progression of the disease. And I will close with a discussion of genetic studies and rationale why the field is so interested in that. I don't think that I need to educate this audience that Parkinson's is a common disease. It's the second most common neurodegenerative disease, with about 1 million people affected by it in the United States, about 10 million worldwide, and based on the most recent literature, as you can see on the slide, it is the most rapidly growing in numbers neurodegenerative disease, probably is the reflection of people living longer, and age is still the most important risk factor for the development of the disease. Now let's dive into therapeutics. Parkinson's disease, I might appropriately say, is fortunate from the, obviously no one wants to get it, but from the standpoint of available therapeutics to treat the symptoms of the disease, compared to other neurodegenerative diseases like Alzheimer's disease and the like, we have quite a wider armamentarium to treat specifically motor symptoms of the disease. And this is a busy slide, that puts the classes of drugs that are available for treatment of the motor symptoms. You can see the word adjunctive therapy. What that means that those drugs are approved for use in addition to levodopa. And the reason why I'm spelling that out and highlighting, because for every person with Parkinson's disease, levodopa, which is dopamine replacement therapy, is really the cornerstone of therapy. And the way I usually say that every single person with Parkinson's at one point in their disease will be on that therapy and will need that therapy. So let's keep going. And before I get into the therapeutics that are in development, I really cannot stress more the importance of non-pharmacological therapy, non-drug therapy. And I know that all of you are educated on the subject. But again, that is really the multidisciplinary spectrum of education on the disease, providing objective information, early introduction of exercise, occupational therapy, physical therapy, speech therapy, social support for the person and the family, nutritional support. And I'm so proud that we as a center really embrace that concept of multidisciplinary care since inception, and still very much pledge and live to that with the help of our collaborators at multiple centers downtown, Shirley Ryan Ability Lab, at all of our region-wide facilities, really having those comprehensive programs. Please use them. And you can see surgical program, which will be very minimal in the scope of my presentation, but it is an important therapeutic tool for the appropriate patients. This is the slide that you probably have seen at a number of talks speaking of the continuum of the disease. And again, the 0.0 on this slide indicates the time when someone comes into the physician's office, is identified to have motor signs of Parkinsonism, and is being told you have newly diagnosed early Parkinson's disease. And that's when the person starts their journey today. And then at some point, and not everyone, develops certain complications of the disease, like uneven response to medications, what is called fluctuations, drug-induced involuntary movements, that is called dyskinesia, some of the what we call non-motor symptoms that again align with more advanced stages of the disease, right? So all of that is to the right of 0. It doesn't escape your view that there is the whole half of the slide on the left that is kind of left blank, right? And that is what we call pre-motor stage of the disease. You also hear the term prodromal disease, but the whole point that I'm alluding to, that not surprisingly, the biology of the disease, changes in the brain, precede by years the first and earliest motor manifestations of the disease. Why am I calling that out? Because we'll return back to that closer to the end of my presentation, where we'll talk about how can we truly prevent the disease. And in order to prevent the disease, we need to go very early on, when the pathology just starts developing and brewing in a person. So now let's move back to the first kind of bucket of my presentation. What do we do well in the therapeutic arena and where we have the gaps? What we do very well is treatment of early motor symptoms. I will not go into every single drug. That is the topic of discussion with your physician, but we have quite an armamentarium of drugs, as you've seen on one of my introductory slides. We do have the options for treatment of dyskinesia, which are both medications and surgical management. Dyskinesia are drug-induced involuntary movements. And if you look at the group of people in the audience listening to the talks, no question about it, I see much less people with all the involuntary movements because we have better treatment options. We have quite a number of treatment options for what we call wearing off, when the medications do not last as long from one dose to the next and people start experiencing recurrence of the symptoms. We have treatment for certain non-motor symptoms, and again, definitely need more, like drop of the blood pressure when someone stands up, which is orthostatic hypertension, depression, hallucinations. And on the right-hand side, it's not a secret to you, there are symptoms of the disease that we don't have good therapeutic options now. I have highlighted balance problems, freezing, which is difficulty to initiate the movements in the motor domain. In the non-motor domain, no question about it, cognitive impairment, which doesn't happen to everyone, usually a sign of more advanced disease, is at the top of the list, but no question about it. Sleep, fatigue, blood pressure changes, all of these can impact someone's quality of life. So where is the therapeutic development? This is a busy slide. This is a multicolored slide that provides you the number of studies across different stages of therapeutic development. So the higher the number is more advanced that study, right? Phase 3 are the studies that are taken to the regulatory agencies, FDA, to seek approval. But really the point that I want to communicate is that the landscape of therapeutic development is getting broader and broader. You can see the dip in 2022. Obviously, that was still pandemic related. This slide did not incorporate the therapeutics of 2023 that I will show you later. So, but really the message that I'm conveying that the field is very rich, the field is very optimistic, and it's really our responsibility to bring these therapeutic developments across the finish line. Now, this is the similar data, but presented in a little bit different view, right? That these are the data from 2023, and out of the active clinical trials, how do they segregate, subdivide, but what symptoms are they aiming to treat? And as you can see, not surprisingly, majority of them are still treating motor symptoms, but again, rewardingly, while the numbers are small, there are studies that are approaching the whole domain of the non-motor manifestations. So let's talk about therapeutics in development, right? I already said that we have a number of therapeutics to treat motor fluctuations, uneven response to medications, but that still is a need. And the therapeutics that are of great interest right now is what is called continuous delivery of the drugs. Some of you are familiar that we have intraduodenal infusion pump that delivers jellified levodopa directly into the gut, but requires a placement of a stomach tube. So there are a couple programs that are developing subcutaneous, under the skin, delivery of levodopa, right? Levodopa is the active compound of Sinemet that a number of you are taking. And I have listed those programs. It's akin to an insulin pump. So the person will need to put a needle under the skin themselves. They carry a small cassette, basically with the drug that is pumping continuously. And the whole idea is that rather than taking the medications, whatever number of times a day, the medication is delivered continuously, which means the medication has a stable level in the body, which means that people should have much less of motor fluctuations. Again, an important milestone is that both of those studies have been completed. They were presented with positive results, and now they're at the stage of either submitting the applications or being reviewed by the regulatory agencies. A lot of patients ask about those. Again, we do not know when they will get approval, hopefully within the next year, but this is the update on levodopa infusion therapy. Another infusion therapeutic is apomorphine, which is dopamine agonist, that has been available in infusion preparation for years in Europe and still in the process of being brought to the United States, but hopefully will be. So again, for people with more advanced disease who need those alternatives to potentially consideration of surgical management for motor fluctuations, those are in the pipeline, hopefully will be coming. right? On the right-hand side are some other novel therapeutics, which are all oral therapies, pills, for motor symptoms, kind of under broad umbrella. So an interesting compound is combination therapy of two previously approved drugs, low-dose dopamine agonist, pramipexole, with long-acting resadulin. And the whole idea is that if you combine two drugs at smaller doses, you could achieve sufficient benefit, but with less side effects and better tolerability of the drugs. So they are submitting, again, application to FDA this year. Another program that a number of you have been asking about is, and you're familiar with the drug Rytary, which chemically is carbidopa-levodopa. So the same chemical compound, but extend long-acting preparation of carbidopa-levodopa. So the same company is developing, quote unquote, a better Rytary, hoping that people will be able to take it twice a day. The studies have been completed. The studies were positive. Not clear whether truly people will be able to take it twice a day, but even 3 times a day, as long as there is steady response to medication, is an advancement. And at the bottom is a new dopamine agonist that is in development. Again, phase 3 studies are ongoing. The field of dopamine agonists have been kind of dormant because for a number of years, about 15 years ago, the whole therapeutic development was around dopamine agonists. Those are pramipexole, ropinirole, reticutin patch, and then kind of the field decided that's enough. So tavapadone is interesting because it acts on slightly different receptors. So it will be interesting to see the results. Let's move on from the motor domain. And I understand that I'm moving very fast, which I usually do, but I repeat it again, that those slides will be, that presentation will be recorded, will be available for you to go back. And obviously I'm trying to deliver to you as much as I can, right? So now let's move to the non-motor symptoms. No question about it, major need. Out of those, I'm highlighting specifically therapeutics that are targeting cognitive symptoms of the disease. And I'm not going to quiz you on all those acronyms and the names, Optinix, Anavex, but the message that I'm conveying to you, that there is keen understanding that we need therapies for those quite bothersome symptoms, and those are being developed. Phase 2 means that they already passed the safety phases of the development, and they're looking at the early signals of efficacy to make the decision whether those therapeutics should move into the phase 3 definitive efficacy studies development. And from here on, I will switch over the gears to the area of my primary expertise, which is development of therapeutics to slow disease progression. A number of you use the word neuroprotective therapies. We try to avoid that because, again, in order to say that word, we need to go in and count the number of cells in the brain. We cannot do that. So the typical terminology used in clinical trials is to slow progression of the disease. And before we dive into specific therapeutics, I really want to highlight this paper. Those of you who've listened to our last year's symposium, I usually bring that paper again and again because I'm really in awe. So 3 people on the screen are 3 people living with Parkinson's disease who are the senior authors on the paper they've been updating on the annual basis, Parkinson's disease, drug therapies, and clinical trials. Kevin McFarthing is a biochemist by background. He lives in Great Britain, and he runs what he calls List of Hope, which is an Excel spreadsheet that is available online. You can Google it. And if you are interested in the most up-to-date, the most extensive compendium of the ongoing clinical trials for any indication in Parkinson's disease, that is the go-to list, not only for you, but we as experts in clinical trials go to the same list. So on the right-hand side is the scheme from this paper. And again, very busy, but this is excitingly busy because it represents all the therapeutics at different stages of the development. So you can see the center of this of the circle is phase 3. Again, therapeutics that are asking one single question: is the therapy effective or not? Phase 2 are preparing for that phase 3, and phase 1 are the earliest studies, first in human safety, readout of the biological indices of success and not. DMT stands for disease modification, slowing progression. ST stands for symptomatic. And again, we're now talking about disease modifying therapies. Look how many therapeutics are there. I promise you I'm not going to cover every single one of them. I have selected just selected few categories that, in my opinion, most exciting and have the most data. So this slide, in one slide, summarizes the disease biology in majority of people with Parkinson's disease. I'm well aware that Dr. Akhtar gave the keynote address today in the morning. So by now you should be experts in synuclein pathology, right? So I'm just cueing this slide to go directly into the next one. And again, he covered that. So what is different from the previous slide to this slide? That the previous slide showed you pathology in someone who is deceased, right? This slide shows you the ability to check whether someone has pathological, abnormally aggregated synuclein in a living person for the first time ever, reliably so. And that is what is called synuclein seed amplification assay. And that is the paper that a lot of you have heard, at least heard about through multiple channels. This is the work by Dr. Andrew Sidorov, representing PPMI data set that we are the participants of, which is the largest data set that has applied synuclein seed amplification and provided the data on how common it is in different subsets of the participants. Again, the reason it's not the first report, but this is the largest cohort that has been published so far. And this is the slide that I know Dr. Akhtar has also shown. So I am doing it for the same reason that, not unexpectedly, majority of people with a clinical diagnosis of Parkinson's disease have synuclein, right? So those are all those blue dots, but not everyone. Why is that important? Because obviously, if you're testing synuclein targeting therapeutic, you don't want to enroll people who don't have synuclein, right? And for the first time, we have the tool to separate those who do, those who don't. And amazingly, people can look exactly the same while their biology is different. And we'll talk about that more. Now let's build on the knowledge that you acquired today in the morning and talk about synuclein targeting therapies. As Dr. Akhtar has covered, synuclein is not a bad guy when it exists in what is called soluble form. It is an important constituent of cells. We still are determining what is the full scope of its work, but it helps the cells to communicate each other. It becomes a bad person when it starts aggregating, right? And it is that aggregated clunk synuclein, simplistically saying, that fills in the cell and prevents the cell to function appropriately. And because it is the signature of the disease, and more than 90% of people with the clinical signs of Parkinson's, that is the primary target of therapeutic development. And as this slide summarizes, there are different places in the cells where you can target synuclein, right? The therapeutics that have been most advanced are what is called monoclonal antibodies that would tie in synuclein that already has left the cells, and simplistically would prevent its propagation to the other cells. So that is postulated mechanism. But there are therapeutics in development that also are reducing synuclein synthesis, and importantly, reducing synuclein aggregation, right? So preventing it going from the normal form to the abnormal form. Now I will show you a couple busy slides. This is the first of them. And again, communicating basically the list of synuclein targeting therapeutics at the top, monoclonal antibodies that are in development. Some of them read out, that will be the next slide. A couple programs have been terminated for multiple reasons, right? And then the other approaches, vaccination for synuclein, for targeting synuclein. So rather than giving passive antibodies, to give a vaccine that will build the body, quote unquote, antibodies against synuclein and aggregation molecules. So again, the field is very active. On the other, which is very exciting. But so far, unfortunately, the front-running large programs both read out negatively. And both of these were monoclonal antibodies. Both of these tested very similar population with a little bit of difference. And here is one slide that summarizes the results of the studies. On the left is a Pasadena study that was run by Roche. On the right is Symposium study that was run by Biogen. Let's cover Biogen study first. Basically, no separation between active therapy and placebo. Negative study, Biogen announced that they're closing the program. On the left-hand side is Roche program. Primary endpoint, very similarly, no separation. But in one of the secondary endpoints that looks specifically at the impact on the motor signs and symptoms, there was separation. Whether that is true to be confirmed, but interestingly, the digital measures that were used in the study as well were very consistent with the clinical measures. And that was the rationale for Roche to continue the program and to roll out what is called not Pasadena, but the Padova study that completed the recruitment, and again will be as definitive as it can be answer to the question whether prasinezumab does have a role in slowing disease progression. So balancing optimism and pessimism, I will show you the next slide that summarizes the story of therapeutic development in Alzheimer's disease. And the message that I'm conveying to you, it takes time. It took Alzheimer's field 20 years to cross the finish line or the starting line. And I suspect that all of you have heard all the excitement about FDA approval for the first Alzheimer's therapy, which is monoclonal antibody against amyloid. Again, I'm not saying that it's easy. And another drug has read out positively and will be going for submission. But again, I'm just reflecting that this is a tough field. It would take time, but there is a lot of optimism. Now let's move away from synuclein targeting therapies to a totally different class of therapeutic development, which is diabetic drugs. And the diabetic drugs specifically that are called GLP-1, standing for glucagon-like peptide 1. So why all of a sudden diabetic drugs in Parkinson's disease? So there are a couple epidemiological reasons. There is increased risk of Parkinson's in people with type 2 diabetes. Again, association versus causality have not been determined. From the potential mechanism, we don't believe that it is diabetes that we are treating, and for that reason, preventing Parkinson's disease. We believe that those class of drugs has impact on inflammation of the cells, obviously has impact on metabolic health, right? Has potentially impact on microvessel changes, has impact on the energy storage of the cells, which is mitochondria. So for all those reasons, these drugs have very solid data from preclinical, from animal studies, and now are extensively studied in people with Parkinson's disease. So, and a lot of you started asking, these drugs are approved for diabetes. Some of these drugs are approved for weight management. And the number of you started coming to the clinic and saying, should I start taking one of the GLP-1 compounds? The most well-known is semaglutide, which is Ozempic, right? But it's not the only one. So this is a summary of the studies that either have read out, look at the study status, or are ongoing. And the important message before I provide you my conclusion and my opinion on that is that every single one of these are phase 2 studies. The only phase 3 study, remember about definitive efficacy question, sufficiently large sample size to be enrolled, is ongoing now. And the results are expected in 2024. So this is slide, a screenshot for that from that hope list. And again, I'm showing it to you not to go drug by drug, but just to give you the reference if someone is interested in more detailed discussion. And now my editorial summary that actually reflects the position statement of the experts in the field, that there is no question mechanistic rationale to study this class of drugs in Parkinson's. Current data has some signal of effect, but is very inconclusive. Results of phase 3 study are pending in the coming year. It's premature to recommend use of GLP-1s off-label in Parkinson's disease. Obviously, these drugs have side effects. These drugs are exceedingly expensive. So that is a major message that I want you to remember. However, if a person has diabetes and Parkinson's disease, it is absolutely reasonable to discuss use of this particular class of drugs with your internist or with your endocrinologist, your diabetes specialist. And the good news are that there have been no new safety signals, concerning signals in Parkinson's. But again, the class has a significant number of adverse events specifically related to gastrointestinal function, and not minor is unwanted weight loss. While the whole world wants to lose weight, people with Parkinson's are actually more prone to lower weight, and again, it's important to maintain. So that is about GLP-1s. Kind of remember semaglutide, Ozempic, right, is the prototypic drug, but not the only one. Let's move on. Another question that comes in the clinic, there have been a lot of, so really, which is centered around, can Parkinson's actually start not in the brain, where we expect it to start, but in the gut? And related to that, if it truly the pathology starts in the gut, are there ways to prevent that pathology spreading to the brain, right? And that's where brain first versus body first hypothesis. It has very interesting data in animal models. There have been no data demonstrating presence of pathology in the gut in absence of pathology in the brain. But this has raised a number of therapeutic questions, specifically if it's gut, is it inflammation that is triggering that propagation of the pathology and the symptoms? How can we approach that inflammation? Can we use improvement of the gut flora and so on? So we don't have the answers yet. But there are studies that specifically looking at gut microbiota, so millions and millions of those microbacteria that are sitting there and their genetic composition. So let's get practical, and all of you asking the question. So should I take probiotics, right? Probiotics are supplements that are not regulated, that you buy in the natural food supply store that improves your gut bacteria, right? So as of today, the answer is if you have constipation, yes. And there are supporting data, right? For slowing disease progression, we don't have data yet. Your next question is which one to take. My answer is we do not know, right? Because the studies that have been done so far are small and need larger studies. What dose to take, again, we do not know. In your question and answer session, I very much encourage you to ask that question to Dr. Bega, who actually has recently completed fellowship in integrative medicine. Are there safety concerns? Again, to the best of our knowledge, no clear concerns. So we just need, as any prescription, we don't send you to take a pill we don't know what, right? We need to provide you more guidance. But I think that it's reasonable for people, just as part of their wellness program, to add probiotic in their regimen if they're interested. Okay. Now let's very quickly go through another important domain. A lot of questions about what about stem cells? So we know the story of stem cells from the 2000s, 20 years ago. And those were negative studies all along, right? However, there is resurgent interest with better technology that actually focuses what is called induced pluripotent stem cells. And those autologous means that they can come from your own body or not, right? So the studies are appearing, and these will be tested in advanced population for legitimate reasons. It's invasive therapy. Some of that requires immunotherapy to suppress immune response to the therapeutic. And the major question is at the bottom of my slide. Will they be effective in slowing progression of Parkinson's, or will they serve as another symptomatic therapy like deep brain stimulation? So the questions, we don't have the answers yet. Another screenshot from the HOPE list, if you want to dive in more into the therapies that are in development, you might see in small print Blue Rock Therapeutic. That is one of the studies that our center will be participating. So definitely our team can give you more information. Let's keep going. And the last topic that I will cover, or what but last, is genetically targeted therapeutics in Parkinson's. Very active area of research. This busy slide really communicates one message, right? That as of today, we treat everyone with Parkinson's as the same. Each of you knows that every person with Parkinson's is one person with Parkinson's. So we need to identify what kind of biology drives the disease in that particular individual. And genetics are a very appropriate entry point, right? Minority of people with Parkinson's will have a single gene responsible for the disease. It's about 5 to 10%. But the two genes that have come to the top of the list are what is called LARC variant carriers. It's about 1.5% of the overall PD population. It might be 30 to 40% in specific ethnic communities, specific Ashkenazi, Jewish, Mediterranean population, kind of all the ethnically communities. And the other one is GBA variant carriers. And again, I will fast forward through this, but I will start with the LARC targeting therapeutics. Very active area of research. You can see that the phases of the studies span from phase 1, which is intraspinal, meaning lumbar puncture, spinal tap delivery of antisense oligonucleotides to quote unquote shut down production of that excessive LARC kinase, to phase 2/3, which is the oral molecule. Again, we're actually participating in both of those studies, and there are some other programs in earlier phases of development. So exciting area, building area. We'll talk about genetic testing in a second. GBA targeting therapeutics, similarly, no successes yet. The front-running study of Sanofi, unfortunately, read out negatively, was well-designed, well-conducted study. But as you can see, there are other programs, and we are participating in a number of them. Dr. Larson is leading the BIAL program. She's also leading the PREVAIL program with gene therapy delivery. So that begets the question, if there are genetically targeted therapeutics, should people know their genetic status as relevant to Parkinson's disease? And that's what this slide is speaking to. Genetic testing is entering the clinic. Obviously, talk to your physician. Genetic counseling is absolutely important because it does have implications for the family. And as of today, US insurances do not cover genetic testing, but there is a number of research programs and direct-to-consumer programs that allow you to have Parkinson's focused genetic testing, and those are listed on this slide. Again, we are participating in number 1 and number 2. Direct consumer testing, i.e. 23andMe, does have very limited panel, but again, I repeat, very limited panel. So be cautious when you do the testing and you say, I don't have genes for Parkinson's, because you don't have only what you were tested for. Let's return back to this slide, and that's where I will be closing my talk of that left side of the panel. And why am I showing you that? Because we started with a notion in the ideal world that we're aiming for, we want to prevent the disease. And in order to prevent the disease, we need to start treating people when they don't have any symptoms in that white space, but when they have the disease biology. And in order for us to do that, we need to all agree that disease starts not when the person comes into my office, but when they are identified to have that abnormal synuclein. And again, right now, sounds to you like a science fiction, but think about your annual health screening. You're being tested for cholesterol. And obviously, you want to know your cholesterol status before you have, hopefully not, a heart attack, right? You are being tested for diabetes risk. And again, you want to know that before you develop obvious signs of diabetes, and so on and so on. So we finally have the tools to move into that phase of ultimately preventive medicine, right? In order to do that, I repeat that we need to shift our thinking, that disease is defined by biology, and the clinical signs and manifestations come much later. They can be called illness, impairment, but the disease is the biology. So that means that we will be telling people who have just synuclein, who don't have any symptoms, you have early stage of the disease. And the bottom of this slide is everything that it enables us to do, to really get into first and foremost, to get into therapeutics early on, right? And we are dedicating a lot of efforts to conceptualize this based on synuclein that we talked a lot about, based on the DAT imaging scan, which essentially gives us the readout of dopamine loss in the brain. But what is most important is that we are moving into that no symptoms, positive biology with a single rationale to ultimately get into the therapeutics of preventing the disease. A lot of work will need to be done. The paper is in submission. That provides the concept, lays the foundation. Right now, it is restricted to research scope, but obviously with maturity of that concept, again, putting the correlation and example of Alzheimer's disease, we anticipate that it will get into the clinic because it's important for people to know. And I suspect that you do want to know your synuclein status. A lot more work needs to be done. As of today, it requires spinal tap, and all of you cringe when you hear about spinal tap, but the technology is developing very rapidly, and I'm an optimist. I expect that in the next couple of years, we will be able to measure synuclein from blood or from skin or from other much more accessible tissues. And now I will close with a call for action. This is for people who do not have Parkinson's, right? Amazingly, loss of sense of smell is present in 80% of people with Parkinson's. It starts a number of years before someone develops motor symptoms. So that's our realistic opportunity to identify those people who might have the biology, bring them into the studies. You know about PPMI study, Michael J. Fox Foundation, and this is one of the initiatives, so spread the word around for the friends and neighbors above the age of 60 if they want to download this QR code and get the smell test by mail if they're interested to participate in the study. And with that, I will close. I know that I have covered a lot, but you are asking a lot of smart questions. And now I will close with two last slides. What should we tell people with Parkinson's disease? Seek expert opinion in a specialized center. Management of the disease is becoming more and more complex and subspecialized. Parkinson Foundation has a lot of educational materials. Inquire about clinical trials. If you don't participate, will not have better drugs. We do have potent medications to treat Parkinson's motor symptoms. Please exercise and please have positive outlook. And this is the acronym that I have borrowed from someone, so I definitely do not claim authenticity. Acronym is MEDS, but stands for not for medication, but for meditation, exercise, diet, sense of well-being, obviously, in addition to appropriate medical management. And this is the very last slide speaking exactly to that concept. Your partners in your journey, be active partners and do your part of the work. Thank you very much. My pleasure.
Thank you so much, Dr. Simuni, for that wonderful presentation. Oh my goodness, so much information, but excellent presentation. We will now hear from Dr. Michael Mercury. Dr. Mercury is a clinical neuropsychologist and the director of neuropsychology and co-director of the Movement Disorders and Neurodegenerative Diseases Center, and was the 2017 through 2020 Douglas L. Johnson Endowed Chair in Neurosciences at Northwestern Medicine's Regional Medical Group. His practice focus is on the memory and other cognitive changes associated with the movement disorders, including Parkinson's disease, and dementias, including Alzheimer's disease.
Good morning, I'm Dr. Michael Mercury, and I'm with Northwestern Medicine, and I'll be talking today about Parkinson's disease and dementia. So what we're going to cover today will be the following. We're going to look at what is cognition, what are the cognitive changes that we see in normal aging. Then we're going to learn specifically about what are the cognitive changes we can see in Parkinson's disease. We'll then talk about mild cognitive impairment, MCI. What is it? How does it apply to Parkinson's disease? And then we'll discuss dementia. What is dementia and how does it relate to Parkinson's disease? So let's start with cognition. We'll start with a definition of what is cognition. Then we'll look at how do we know what's normal? And then we'll give you an example of how do we actually measure cognition. So we divide the brain up into different geographic areas, and those geographic areas contain certain cognitive or functional domains that we talk about. So if you look at the picture on the left, that's the frontal area of the brain, and that's executive functioning. So executive functioning, that's your things like attention and concentration, organization, judgment, decision-making, and so on. So that resides there. If you look at the picture on the right, we're looking at the back of someone's head. The left side has people working in cubicles, and the right side, you've got people enjoying a beautiful day at the park. So the left side of the brain is our verbal brain. So understanding what I'm saying right now, me speaking, that's all language functions. That's on the verbal side of the brain. And also verbal memory is on that side of the brain. So remembering what I hear, remembering what I read is going to be in that side of the brain. The right side of the brain, we call it the visual spatial side of the brain. That helps us interpret what we're looking at. It helps us figure out where our body is in space without having to think about it. So when you walk through a doorway, you just walk through the doorway. You don't worry about how many inches you are from the right side or the left side. That's all the function of the right side of the brain. It also contains visual memory. So for example, if we're driving somewhere and we've got some geographic landmarks in our head, that memorization of visual stimuli is all on the right side of the brain. So that's how we look at the cognitive domains of executive functioning, language, memory, and visual spatial abilities. Now, in reality, the brain is more complex. There's more of a network functioning, but this helps for looking at the functions of the brain and how the brain interprets the world around it. Now, how do we measure that? The way we measure it is traditionally with paper and pencil measures. They actually look like school tests, but you can't really flunk them. They just tell us, is your memory, I'll use memory as an example, is your memory age appropriate? And we're slowly going into more computerized tests, which have the advantage of being quicker and giving us even more information. Now, how do we know what's normal? It's because of volunteers, and the volunteers I'm most familiar with are out of Seattle. The Seattle longitudinal study started in 1956. The oldest volunteers right now are 88. Isn't that amazing? Some of the same people have been doing this since 1956. And if you look at this curve here, so on the bottom line here on this axis, you've got age. On this one, you've got scores. So as it goes up, that's a higher score. The good news is we actually do get wisdom as we get older. So don't let young people tell you you don't get wisdom. You do get wisdom. Look at this right here, starting around age 53, you look at inductive reasoning. It's at its peak. Now, some things decline over time, like our processing speed. You see the green circles here. Those go down over time. So those are how we know some of the natural changes that occur with aging. Another way we know is when we have a new test that comes out, we give the test to people of different ages, and we look and see, are they representing normal aging according to the test results? So we use that as a very important factor in interpreting tests. Now, let's look at the cognitive changes we can see in Parkinson's disease. So once again, we're going to look at the 4 domains of executive functioning, language, memory, and visual spatial. Now, if we go back to what Dr. Parkinson said in his treatise, he said, if you look at the last 3 words, intellects being uninjured. So he did not see the intellectual changes or the changes in cognition that we're going to be talking about today. And part of the reason for that is they can be very subtle and they can be hard to detect, but they are there, and knowing about them is important. So we're going to look at the trajectory that we see in Parkinson's disease. So on the left side, where you've got the white column, that's where Parkinson's disease is kind of below the surface. It's percolating, but we don't have any motor symptoms at all. And as it shows here, this is a timeline. So this is timeline 0. As many as 20 years before, someone might have new adult onset constipation. Somebody might have dream enactment, this RBD, REM behavior disorder, as many as 10 years before. So there are certain things that can occur that we might not associate with Parkinson's if we think of the motor aspect of it. And in addition to those, we can sometimes also see some cognitive changes. I've had the experience of, unfortunately, seeing younger people come in with Parkinson's who said that 5 years before they were fired from their job because they were only able to manage 2 projects and they had always managed 5 projects at a time. And at that time, that was probably the slowed processing, which I'll talk about in a little bit, that can occur in Parkinson's, but they were not aware of that. And so unfortunately, they got fired. Another thing I want to talk about, I'm going to talk about some of the changes we see, but I want you all to be aware that not everybody gets these cognitive changes. So let's look at the next slide. So we see these cognitive changes in about 30 to 40% of people with Parkinson's disease. So as you've probably all heard from your movement disorder specialist, everyone with Parkinson's disease is very different. That's true for the motor. That's also true for the cognitive. And another important fact is these cognitive deficits do not constitute a dementia. And I'll be defining a dementia in a little bit. So let's talk about what are some of the cognitive changes we can see in the different domains. We start with executive functioning. We do see some impairments there. Now, simple attention, just paying attention, is usually not affected. And that's also why sometimes these problems, these cognitive problems can be missed, because the person seems to be doing just fine. In reality, though, one of the things that I see with a lot of patients is processing speed becomes very slow. So it doesn't affect intelligence, but let's say I go to add some numbers in my head, takes me longer to do that. The example I gave you with a gentleman who could only manage 2 projects instead of 5. Language is technically spared. It's not affected. And the way we're defining language here is if we look at something, look at a picture and say, you know, this is a tulip. And we look at a picture and say, this is a house, that sort of thing. But what can be affected with language is word finding. And that's actually an executive functioning problem, right? It's the slowed processing that gives me trouble finding the word. It's not because I have a language deficit. Memory has a very distinct type of change. What we can see there is difficulties with remembering, not forgetting, but difficulties with remembering. I'm going to give you all an example that we experience almost daily. You walk into a room and you can't remember why you went into the room, right? And usually if you retrace your steps, you're like, oh, I went in there to get the Scotch tape or something. That's an example of a retrieval memory problem where I don't recall, I don't remember the reason I went in at that moment. Or you're watching a movie and you really like that movie star and you can't remember his or her name. Comes back to you later, somebody gives you a hint about a movie they were in. So in both cases, I know the information, it's stored in my memory. I'm having trouble retrieving it at the moment I want to know it. So it can, in the real world, look like I've forgotten. It can feel to me like I've forgotten. But what we see with Parkinson's is usually if you give extra time or if you give somebody a hint, the information is going to come back to them. And then visual spatial. So visual spatial tends to be impaired, and I'm going to give you a few examples of that in just a moment. So I'm going to cover each of these changes in a little bit more detail, just so that you're able to understand it better. So let's start with executive functioning. So executive functioning, again, that helps run the whole brain in a sense. So that's our concentration, our focus, our multitasking, our ability to start and to follow through on a task, our planning, our organization, our processing speed, our problem solving. So these are very important functions. And like I said, the one that really stands out tends to be the slowed processing. Although I do have a lot of people saying they can't multitask, but I would argue that probably the multitasking is related to the slowed processing. Now, I'm going to introduce on a couple of these slides 2 factors that are very important that we have to pay attention to when we look at the cognitive functions, because these cognitive domains are not by themselves the only influence on how our brain works. We know that in about 40% of people with Parkinson's, we can see anxiety, and almost an equal amount we can see depression. And I'm going to talk about how those can affect each of these domains. So starting with executive functioning. Attention concentration can be affected by anxiety. So for example, I'm talking to you right now, and if I'm thinking, oh, I should have made this phone call before I got onto this talk, and I should have done this, and I should have done that, I'm distracted. Similarly, if you're telling me something and I'm thinking about all these things I could have, should have done, I might only actually process part of what you're telling me. Maybe I get 40% of what you said. It's going to look like I forgot. It's going to feel to me like I forgot later on, but in fact, I was distracted. So anxiety, the disease of anxiety, can interrupt executive functioning. So we can have poor concentration that can look like memory issues. If we look at initiation, boredom, things that look like apathy, depression can be a factor there because depression can affect motivation. Depression can affect sleep, and sleep can affect our ability to do things, our motivation. So a lot of these things are intertwined. And towards the end, I'm going to go into a little bit more detail about factors we should look at when we're examining these cognitive domains, because they are not by themselves causing or helping the brain function, they are influenced by other things. Let's look at language next. Like I said, language is spared. The problems we see with language are slow processing. So we might hear a slow speech pattern when someone is speaking. We might see some slowness of thought. People often tell me that when they are in a group of people in a conversation, often by the time they formulate what they want to say and give their opinion, the group is on to another topic. And so they just don't talk. They become very isolated in this group. And so what I'll often recommend, particularly if it's a group of really good friends or relatives, is maybe as they're about to move on to a new topic to say something like, hey, Michael, did you want to give an opinion on this as well? And it can work really well because it gives the person a chance to contribute something. And then the word finding difficulties that I talked about. And once again, this is all worse with depression. Depression will amplify the slowed processing. So we really need to be aware of that and to treat depression and anxiety, which are very treatable. What about memory? Well, let's find out, what do we need to do in order to learn something? I mean, to have memory. We have to learn something first. Memory itself is the persistence of that learning, and we have to be able to retrieve it. Let's go to learning, and let's go back to anxiety, which can affect my attention concentration. So that information, part of it may never make it into the memory storage areas. Then we've got memory, and then we've got to be able to retrieve that memory. We need to get to that memory. And I gave you the examples of walking into a room, remembering somebody's name. So retrieval is what becomes problematic with Parkinson's disease. And being aware of that and knowing I might need some extra time, or I might need a cue, or just knowing that post-it notes are actually going to help me to remember, that's important. But once again, depression causes the same type of memory problem, and so can actually exacerbate memory changes in Parkinson's disease. Here's an example, and some of you might have had some tests where your doctor asks you to remember 3 words. And people with Parkinson's might remember all 3 words, but on some occasions they might not remember all 3 words. They might remember 0 words. And if you stop there, it looks like they're forgetting. Let's say one of the words was window. What we know with Parkinson's, if we say something like, well, you know, it's parts of a house, usually they're going to remember window. Or if they don't remember window, if we say, well, was it a door? Was it a floor? Was it a window? And they'll recognize that it was window. So retrieval memory problems are what we see with Parkinson's disease. Very important to be aware of that, because if someone becomes very frustrated at their difficulty, it's going to be even harder to retrieve that information. So the last cognitive domain is the visual spatial. Parkinson's disease can actually interfere with contrast sensitivity. And you see the picture on the right, someone's driving at night and the white lines are not as sharp and distinct, often with Parkinson's disease. It can also affect just being able to tell differences in shades of color. So if you're picking out color in your room. There's some diminished color vision with Parkinson's disease as well. Understanding nonverbal cues can be a problem. So for example, we know with Parkinson's disease, sometimes the patient gets masking of the face, but in addition, they can have difficulty in interpreting other people's facial expressions or nonverbals. So I've had couples where, the spouse will get really angry at the other spouse and give them a really, mean, angry look, and the patient doesn't react at all, which of course makes the spouse even angrier. And we let them know about this. They might not be understanding your nonverbal cue there. You might want to say, I'm really angry with you right now, and reinforce what you're trying to show through your facial emotion. So that's important. And then spatial awareness. So I talked about, where your body is in space, like walking through a doorway, but your car is an extension of your body. And I've had patients over the years say that sometimes when they're driving, they notice that they drift a little bit to the yellow line, and it's harder to stay in the middle of the lane. So that's important. Now, my picture here, you'll notice they're all kind of on the wrong side. It's from England. I couldn't find an American picture of it, but that's why this guy is on, they're on this side of the road going in this direction, but he's going towards the yellow line. So that's why I used that. And guess what? Depression. Depression makes contrast sensitivity worse. So who knew, right? We think of depression as a mood disorder. It affects vision, it affects sleep, it affects appetite, et cetera, et cetera, et cetera. So just please be aware of anxiety and depression, very, very treatable. Talk to your movement disorders doctors about those. Those are going to help your cognitive domains be at their best. And that's what we want, of course, best functioning possible. So I'm going to go on now to mild cognitive impairment, MCI. What is mild cognitive impairment? How do we manage it? And if we look again at this timeline, we're now into the area where we do have the motor symptoms. We're a few years into the disease, and we get this change called mild cognitive impairment, potentially. Again, remember, not everybody gets this. So to understand mild cognitive impairment, or MCI, we have to understand two criteria, and these two criteria have to be met. Criterion number one is they have to be impaired on neuropsychological testing. So what you're seeing here in red is the normal curve, and what it shows in the dark red is that most people would fall within 1 standard deviation on testing. And then you've got people going all the way to 2 standard deviations. So everything above is within the normal range. When we get here below the normal range, about 1.5, as it says up there, that's when we say we've got some impaired performance. So right about here. And we say that that's based on age. Some tests it's age and education. So it just depends on the test. But if they're 1.5 standard deviations below normal on formal neuropsychological testing, that's criterion number one. Criterion number two is that they're functioning okay in the real world. So look at this part of the slide. That's called the instrumental activities of daily living, IADLs. So the instrumental activities of daily living are things like managing your medications, things like transportation, managing finances, and so on. So being able to run a household. So what happens in this situation is the person is able to manage these functions even though they're having some cognitive problems. So say I've got some memory concerns. I get the testing, my memory is in the impaired range. I'm not forgetting, remember, just having trouble retrieving information. So I need reminders, I need hints. So I've got a smartphone, and every day at 9 o'clock, at noon, at 6 o'clock, that smartphone goes off and tells me basically to take my medication. It's my reminder. And as far as managing my finances, I have a whiteboard like you wouldn't believe with the month on there, and I've got every doctor's appointment, I've got when bills are due, what I'm going to pay, and you know what? 90% of the time I'm paying my bills just fine. So that would be an example of mild cognitive impairment. I have some difficulties. It's seen on testing, but I'm managing okay in the real world independently. Now, MCI, or mild cognitive impairment, by our psychiatry colleagues is called minor neurocognitive disorder. So I just wanted to let you know that. We tend not to use that in our center. So the gist of it is the cognitive deficits interfere, but they do not interfere with independence. That's the key. How frequently do we see this? There was one sample, 7,000 people, 40% of the sample had MCI. When we have something like MCI, we're always going to be looking for reversible causes, making sure it's not another disease, such as a confusional state, which we call a delirium or a stroke, again, depression. And we're also going to look for reversible causes. So again, going back to that anxiety and depression, sleepiness, other things that might cause us to think the person has MCI, or things that would make the MCI seem worse than it actually is. So how do we manage MCI? So remember, the person is able to come up with strategies. So let's help them. A wonderful service we've got at all of our movement disorder centers is LSVT, Lee Silverman Voice Therapy. And that's through our speech therapists that help people to speak loudly by projecting their voice. There's a physical version of that where exaggerated movements are used. It's called, and that's by our physical therapists and our occupational therapists. Exercise is wonderful. Some of you, do boxing and different exercises like that. And the good news is you're getting a twofer with the exercise, because we also believe, and this is based on some UCLA studies that have been ongoing for years, that good exercise will also prevent or at least delay dementia. So you're not only helping the Parkinson's, you're hopefully preventing a dementia as well. Cognitive rehabilitation is available from speech and occupational therapy. Our occupational therapists are wonderful. I had just recently a gentleman, and he's a computer guy, and his keyboard's no longer working for him. So she's working to find a better keyboard for him that works for him. And then Shirley Ryan Ability Lab does a terrific job with people who are still working with Parkinson's. There's a peer support group, which is just excellent, and it's virtual. And they can do a vocational evaluation if somebody has to modify the type of work that they're doing. So they just do a great job. So those are the type of things where we can help people identify strategies, work on them, keep them as healthy as possible, and work around the difficulties they're having. So that's MCI. Let's go on now to dementia. And we'll define dementia. We'll talk about types of dementia. And we'll then zero in on Parkinson's disease dementia and talk a little bit about Lewy body dementia. We'll talk about some symptoms that can be misdiagnosed as dementia, and then we'll talk about management. So looking again at this timeline, we're now about 10 years into the disease. Again, everyone's different. We've got other difficulties going on, but that's where we might start to see dementia. Dementia also has another term that the psychiatrists use, which is major neurocognitive disorder, just so you're aware. Now, like MCI, dementia is a concept, it's not an actual disease. There are certain diseases that fall under dementia. So we call it an umbrella term, because if somebody has a dementia, what we say is, what is the most likely underlying disease causing the dementia? Now, we go back to the two criteria that we had looked at with MCI. So with MCI, remember, the first one was the same, impaired neuropsychological testing. But here's the difference. Look at the second one. Now we've got impaired instrumental activities of daily living. So the person now is not able to function independently. The timer on my smartphone goes off, and half the time I don't remember to take my medications. That whiteboard I was so proud of, I don't fill that out anymore. That's too much work. Or I fill it out incorrectly. So they just turned off the electricity in my home. So it becomes now problematic. I need somebody else to step in. So that would constitute a dementia. Those wonderful strategies I was using before on my own, I can no longer use them to compensate. So when we say dementia, those two terms are met. And then we ask, what is the most likely underlying disease? So that's where the disease comes in. Dementia is just this umbrella term. If you look on the left, you've got Alzheimer's. So Alzheimer's is anywhere from 50 to 75% of dementias. Now, some of those are a combination of Alzheimer's and another disease, like a vascular disease. And then some of those are pure Alzheimer's. But Alzheimer's is the most frequent cause of dementia. And just to give you some quick statistics, 75-year-olds in the United States, about 25% of them have Alzheimer's. 85-year-olds, it's almost as high as 40%, depending on the study you look at. So age is the biggest risk factor for the type of dementia due to Alzheimer's. Vascular disease and Lewy body disease Some studies indicate that they're about the same as far as how often we see them. In this cartoon, it puts vascular first. So vascular disease is anything affecting the blood vessels. So that would be heart disease, stroke, diabetes, untreated sleep apnea. Anything affecting my blood vessels could cause a vascular disease. Lewy body we'll talk about in a little bit more detail, but it involves some Parkinsonism and some other factors. And then one that's not as common is frontotemporal dementia. And you might have heard of in the media over the past few months about Bruce Willis, and he has a frontotemporal dementia where he's losing his ability to speak. And so he has a subset of that called primary progressive aphasia. So that's not as common. Now I'm going to use Alzheimer's as an example of how we rate whether a dementia is mild, moderate, or severe. To look at mild, moderate, or severe, we go from the instrumental activities of daily living, that's for functioning in a household, to activities of daily living, which is taking care of one's body. So we say it's mild if I can pretty much take care of my functions for my body, like bathing, eating, mobility. Even if I'm using a walker, that doesn't matter, that sort of thing. So really very, very little is interfering with that ability. Now, all of a sudden, if I'm no longer able to take a shower on my own, even though I got the shower chair in there, I'm still unsteady, I really need somebody to literally be there with me. And maybe I'm so unsteady as far as mobility goes that the walker's no longer enough. I need somebody there kind of spotting me and making sure I'm safe. Then we get more into the moderate as those things increase. And then severe would be all of the activities of daily living being affected. So that's how we rate it as mild, moderate, or severe. We would do the same thing with Parkinson's type dementia, anything like that. The 3 R's, though, of a dementia workup are, can we reverse it? Can we rehabilitate it? Or can we somehow reduce the impact of it? So that's what becomes important. Give you some examples now. And this is going back to, remember at the beginning, I talked about how anxiety and depression can interfere with the different cognitive domains. These are the other areas that can interfere. So first of all, we want the person as healthy as possible. So we bring in the team, the primary care physician, the movement disorders neurologist. If they're over 70, maybe a geriatrician, maybe someone who's specializing in dementias and things like that, like a behavioral neurologist. We check their labs. Low vitamin B12 can cause memory problems. Give them some B12. That should improve the memory. We look at their medications, and particularly the over-the-counter. The bane of my existence are the sleep medications, Tylenol PM, Advil PM, Bayer nighttime, Z-Quil. All of those have another medication in them that can cause constipation, problems with walking, and confusion or memory problems. So in other words, it can add to the problems that the person has due to the Parkinson's. We want to address physical problems. Pain causes attention and concentration problems. Constipation, same thing, can also be life-threatening. We want to take care of the issues we can take care of. Hearing, like anxiety, can interfere with learning. So if I have a hearing problem and I'm focusing so much on what you're saying, I'm looking at your lips, and I missed a couple words from that first sentence, I'm distracted by the mechanics of trying to hear you. I miss the information you're trying to give me. It's going to look like I'm forgetting. It's going to feel like I'm forgetting. It's my hearing problem. And to make matters worse now, there's been a couple studies out, these are kind of preliminary, showing that untreated hearing loss might increase our risk of dementia. So definitely get that taken care of. Sleep problems. Are we getting good restorative sleep? And one of the things I always want to rule out is sleep apnea. Sleep apnea is not only annoying because it keeps the spouse awake, the bed partner awake, it's also potentially a cause of stroke and death. Not good side effects. So you want to be careful with that. Alcohol. As we grow older, we can have less alcohol, of course, and they've changed some of the recommendations on that, but also with certain medications, alcohol can be a problem. Cannabis use. I was just at a conference a few months ago, and we had a pharmacist go over the studies on marijuana. And as you know, it's been illegal to study that for years. And guess what? The studies were terrible. So a lot of the quote unquote approved uses are based on anecdote. So be very, very careful. And then, of course, depression and anxiety. So let's talk about what is Parkinson's disease dementia. So we know the changes we can see are executive functioning and visual, spatial, some retrieval memory. Those are kind of normal changes we see in Parkinson's. But when it evolves into a dementia, what we've got is the executive and the visual spatial deficits are interfering. They're so great that they're interfering with the ability to be independent. And then usually we get some neuropsychiatric symptoms like hallucinations becoming more prominent and so on. This represents about 3.6% of all cases of dementia in the population. So just thinking of the different ones that I showed you in that one cartoon. What are some of the risk factors? An older age, longer disease duration, older age at disease onset, and then the severity of the Parkinson's itself. And once again, the two things that are really going to stand out are the executive dysfunction and the impaired visual spatial function. Memory problems, you still have your retrieval memory, but those are not prominent. It's really going to be the executive and the impaired visual spatial. And then you can have some of these neuropsychiatric changes like apathy and so on. Now here's the hot button issue. What about driving? So driving is often one of the earliest issues that needs to be confronted, and people mistakenly think that driving might be a concern because of the motor aspect of Parkinson's, whereas actually it's the cognitive and visual deficits that can cause problems with motor impairment. On the cognitive side, it's executive functioning, it's the ability to process information quickly, it's the multitasking. That's what can be a problem there. Let's say I'm sitting at an intersection, the light turns from red to green. Am I going to go right away? Well, I got to judge whether there's anybody who's planning to go through that red light on the other side. What about that kid on the bicycle on my left here who's about 8 years old? Is he going to go when he's not supposed to and try to cross the street? I need to process a lot of information. I need to do it quickly. So multitasking and processing speed can be very, very important in judging whether someone is safe to drive. And the thing is, with the dementia, by definition, it gets worse over time. So I might be safe to drive now, but am I in 3 months? So it has to be really looked at and discussed. Now, what's the difference between Parkinson's disease dementia and Lewy body dementia? The definitions are this. If you look at the first one, Parkinson's disease dementia, the dementia develops after the motor disorder. So you saw on that timeline, it was about 10 years into the motor problems. So we would call that a Parkinson's disease dementia. If the dementia occurs simultaneously with the motor problems, then we're thinking more dementia with Lewy bodies. There's a more formal definition, of course. Some people feel that the diseases are actually the same. They're more on a continuum. So more research is needed on this, but that's the basic difference by definition that we use. I want to go over just briefly what are some symptoms that could be misinterpreted as dementia. And a few years ago, a group of us went to a facility and we tried to train the staff, which included the people who served meals, the nursing staff, janitors, everyone, about what are some of the things you might see in Parkinson's, and just making sure that they weren't misinterpreting people as having a dementia. So we know verbally you might get the quiet voice. You've got the psychomotor slowing, so they might be slow to respond if you ask them a question, the small writing. And then if you look at the nonverbal, they might have kind of a masking of the face where they don't show expressions really well. And so they might look like they're just kind of staring and not really listening to what you're saying. There can be a reduction in body language. As I said earlier, they might not understand the nonverbals of the person and so not react the way you would expect. And so people sometimes can mistake that for a dementia, that the person's not able to pay attention, to participate in a conversation and so on. So really important to let people know you just need to give extra time, speak to people. They don't have a dementia. So that's important because that can be misinterpreted. So issues in late Parkinson's disease dementia. One is you want to get your affairs in order. You need a power of attorney for healthcare. In fact, everybody 18 years and older should have a power of attorney for healthcare. I am very passionate about that. Get a power of attorney for healthcare. You really need it. Now, in addition, if you have a chronic disease, you should also get a power of attorney for finances. And you and your family and your doctors should talk about important issues like driving. Can the person live alone? Can they manage their medications? It's okay if you can't and someone has to help you. It's more important that you take the medications properly. And then finances. So how can we treat this dementia? We don't have a disease-altering treatment. We can treat some of the symptoms. The changes we can affect are modest, but worth trying. Again, some people might benefit more than others. So one is the cholinesterase inhibitors. Acetylcholine is an important chemical in the brain. There's rivastigmine. If you look at the data, though, only 15% of people benefited, but 15% benefited. Donepezil, possible benefit for executive functioning. And then another one that has a different functioning, memantine, showed some improvement. Here's the problem. We sometimes will see an improvement on a test. It's statistically significant, but it's not really making improvement in the real world. So everybody has to be kind of checked over to see whether or not adding a medication is a good idea and whether the potential side effects outweigh the potential benefits. So finally, what do we need to do? Wellness in an advancing dementia. Meaning, purpose, and dignity. I'm just going to read this to you because I think this is really well said. We all need meaning and purpose in our lives. Just because a loved one has cognitive problems does not mean that they cannot contribute and be connected to others, even if the job they do is imperfect. We all need to be treated with dignity. So if I can participate in the kitchen and maybe I'm not doing a real good job at what I'm supposed to be doing, but I'm there, I'm with you, I'm with the family, I'm having a good time, I'm safe, then I should do it. I call that the principle of partial participation. It's a very good thing. So in summary, what did we talk about? So we reviewed cognitive domains. We reviewed some of the cognitive changes in normal aging. Remember, you do get wisdom. We learned about the normal cognitive changes in Parkinson's disease. So if you have those changes, be aware of them, get some strategies. They do not constitute a dementia. Then we learned about what's mild cognitive impairment. What does that mean? We talked about the two criteria, how someone does in neuropsychological tests, and then whether someone is able to function independently in the real world. And then finally, we talked about dementia in relation to Parkinson's disease, or Parkinson's dementia. So I hope that was helpful to everybody, and thank you very much for your attention.
All right. Thank you so much to Dr. Mercury for such a wonderful presentation. And thank you to all of our presenters today. I think there was so much wonderful information that we were able to share with you all today. I hope you feel the same. We will now transition to the Q&A panel, and we're going to take some questions from the audience. So I'm going to start by introducing today's panelists. We have Dr. Danny Bega, Dr. Tanya Simuni, Dr. Rizwan Akhtar, and Dr. Michael Mercury, all of whom you have heard from today. And I will begin by asking some of the questions that have been coming in throughout the presentation, and you can continue sending your questions to the Q&A box in your Zoom control panel. We have truly had an overwhelming number of questions today. which is wonderful. And we're so happy that you all want to learn more and are engaged in the program. So we will do our best to get to as many questions as we can. And I know that some of the questions were addressed in the presentations today, but we will send out in our recap email some further questions. We will respond to them, some of the questions that we were not able to actually address in the Q&A panel. So moving on, my first question is actually for Dr. Danny Bega. Dr. Bega, are DAT scans recommended to differentiate PD from essential tremor?
Can you hear me?
Yes.
Thank you. So let me start by saying what a DAT scan is for those who don't know. It's a really interesting tool that we have available. It's FDA approved. It's available at a lot of medical centers, and it's a scan where a dye is injected through an IV. That dye binds to some receptors in the brain, in the areas of the brain that produce and release dopamine. And what we can see on that scan is just generally whether those areas of the brain are present in normal amounts and taking up this dye. And so if it is taking up the dye normally, that tells us that the cells are there. They're able to take up that dye. In someone with Parkinson's disease or another degenerative disease where they're losing neurons in that part of the brain relevant to dopamine, you lose some of those cells so they don't take up the tracer normally. So in other words, that scan can tell us if someone has a normal amount of those dopamine producing cells or not. It's really helpful to us in some situations, particularly if someone has really mild, subtle symptoms of Parkinson's. We're not really sure if they could be normal or if they're really abnormal because they're so subtle. That could be a good use of a DAT scan to help us kind of be convinced that there is actually loss of these dopamine cells. The question was about distinguishing Parkinson's disease from essential tremor. That's actually the main indication that scan is approved for. It's a very good use of that scan in distinguishing when someone has a tremor, whether it's Parkinson's tremor or a benign essential tremor. The scan can distinguish between those two. The scan is not needed in everybody who has Parkinson's disease. For the most part, it remains a clinical diagnosis, and in the majority of people, we're able to make the diagnosis based on taking a good history and doing a good exam and ruling out other things. But in some cases, it is a really helpful tool when there's uncertainty. One other thing is it does not distinguish between different causes of degenerative Parkinsonism. So it won't distinguish between Parkinson's disease and Lewy body dementia and PSP and some of these other atypical causes, unfortunately.
All right. Thank you so much, Dr. Bega, for answering that question. My next question is for Dr. Simuni. Dr. Simuni, can you please speak more about the new proposed staging of Parkinson's disease?
Yes, I can. So Dr. Akhtar has done a terrific job introducing the whole concept of biology and speaking a little bit about staging. I will speak about it, but very briefly. So let's start with a concept and really reiterate what Dr. Akhtar has said, that first of all, you need to define the disease, right? And transformative, what we are proposing is that disease is defined by biology. So when the person does not have any symptoms, but they have the biological substrate of the disease, which now you're educated, synuclein pathology, they have the earliest stages of the disease, right? And the idea is not to scare people, but to catch, quote unquote, the disease in its earliest pathology-based state to ultimately start prevention therapies there, right? And then as pathology progresses, as the biology progresses, people will develop dopamine dysfunction, as Dr. Bega has just alluded. And then with those attributes, people who come to us in the clinic, as Dr. Akhtar has indicated, 90% of them who are given diagnosis of clinical diagnosis of Parkinson's disease do have synuclein pathology, do have dopamine dysfunction, and further staging. So the first stages are based strictly on biology, and then further stages are not just based on presence of the symptoms, but based on the degree of functional impairment, highlighting that what is important for the patients and for the regulators who approve the drugs is not what I see, but how the person feels with their symptoms. So that is conceptually the staging. The staging that is proposed now is for research domain. It's not ready for clinic. There are a lot of field, quote unquote, field testing in the real world testing that we need to do. As of now, it is positioned that it is an important tool to position the therapeutic trials for better success, because we'll be recruiting more homogeneous population. We'll know what pathology drives their disease, and obviously it's essentially important for successful therapies. At some point, we envision that it will come into the clinic, but again, more data necessary.
Thank you so much, Dr. Simuni, for that excellent explanation. All right, my next question is for Dr. Akhtar. Dr. Akhtar, can you speak specifically about the skin testing method, like samples from the leg or the back? Is this available? Is it accurate? And can you speak to its purpose?
Sure, I'd be happy to talk about that. I did not talk about that much in the talk today. So that refers to a test that's marketed or at least advertised as the SYN-1 test. It is a test that involves collection of skin from 3 sites on the body. The skin collection is a small punch that's similar to what a dermatologist would do if you present there with a mole or a lesion of some kind. The skin punches are sent to a central lab where the skin is examined for signatures that go along with Parkinson's disease. And this has been in development for a number of years now, and I think about 3 or 4 years ago became more in the limelight after a paper was published that showed you could see these signatures in the skin. And biologically, it does make sense to look at the skin, even though it may not first think about that as a good place to look at brain pathology, but it turns out that there are a number of small nerve endings in the skin that we can examine if you take a piece of skin, and those nerve endings seem to be affected by similar processes that happen in the brain. Obviously not the same, but similar. So what does that mean? Do we need to use this test? So this test at the moment is only available through, as far as I know, one company. That company has not applied for an FDA type of exemption for that test. And it is a test that does involve you're coming to the clinic to have these skin biopsies collected, which is not something that all neurologists are trained to do. And so at the moment, we are still in this transition to see whether or not we can really use this test clinically or not. The data suggests, has been published, suggests that the skin test can separate patients who are healthy from patients who have Parkinson's disease. It does not currently clearly show a difference between people who have Parkinson's disease and other synuclein disorders like dementia with Lewy bodies or multiple system atrophy. And again, because it's only available through one place, it hasn't really been replicated the way we like to see. So at the moment, we are actually talking about potentially sending those from the clinic, but it's not something that we are doing currently, although I think over the next year, we'll have more to say about that.
Wonderful. Thank you so much, Dr. Akhtar. My next question is for Dr. Mercury. Dr. Mercury, can hearing loss, mild to moderate, complicate or confuse testing or determining any sort of cognitive decline?
That's an excellent question. Yes, it definitely can. So most of our tests use verbal instructions. And so as a result, if someone does have a hearing disorder, and particularly if it's mild and we're not aware of it, they might misinterpret directions. We actually have what's called a pocket talk in the center so that a person can put on headphones. There's a microphone if we do detect that they have some hearing loss. I try to elicit that during the interview. Often the patient's going to say no, and you've got the spouse going, yes, he does have hearing loss. So that's often helpful. But that's definitely a problem. And similarly in everyday life, one of the things we know that helps to, we believe, delay any sort of a dementia is being socially involved. And people who have hearing loss often will be very reticent to say anything in a group conversation, particularly in a restaurant where there's a lot of background noise. So hearing is huge, not only in the testing environment, but also in the real world. And we do try to account for that and note that a test might not be accurate because of the hearing problem.
Excellent. Thank you so much, Dr. Mercury. I just want to announce to all of our attendees that we are going to extend our Q&A panel to 1:10 so that we have a full 30 minutes to answer as many questions as we can. Like I said, we were overwhelmed with the amount of excellent questions that we had. I am going to take the next question. Somebody asked a question about deep brain stimulation as a therapeutic, and I want to encourage you all. Our center out at Central DuPage Hospital is actually hosting an event on October 17th talking all about deep brain stimulation. Dr. Mercury is going to be a part of that panel, and they're going to be including panelists that have actually received deep brain stimulation. So if you are curious to learn about deep brain stimulation, I encourage you to register for that event. You can register for that event by going to nm.org/Parkinson's, and there you will see a button that can redirect you to registration for that event. That event is being held in person, but due to an overwhelming response to that event, it is being extended as a virtual component. So the in-person event has been filled, but you are welcome to join virtually for that event. Okay. My next question is for Dr. Danny Bega. Dr. Bega, how often does dyskinesia develop in patients with Parkinson's disease? Could you speak to what percentage that would be?
Sure. I'll just define dyskinesia real quickly. It's dyskinesia is a term for abnormal involuntary movements that people get not from Parkinson's disease, but related to the treatment that we use to help with the slowness and stiffness, primarily the levodopa treatment. While levodopa works very well for treating slowness, stiffness, the general common features of Parkinson's, it can come with the cost of causing some excessive movements, which can look like fidgeting or dancing or rocking. And it tends to occur when the levodopa, for most people, when the levodopa is at its peak, is when they tend to have that complication. Sometimes it's the family members who are more bothered by it or notice it first. Sometimes it's surprising that the person experiencing the dyskinesias may not be as aware of it. And at other times it can be bothersome to the person living with it. And it is common. What we know is that the longer someone has had Parkinson's, the more likely they are to have it. And going along with that, the higher the dose of levodopa that's needed. And so it's the two things together that make it more likely. By about 5 to 10 years with Parkinson's, we think about 70 to 80% of people will experience some dyskinesia. But that is a spectrum. It can be very minor and subtle, all the way to more bothersome in that amount of people. By over 10 years, it's probably closer to 80% or so of people that will have some dyskinesia. Luckily, there are ways to manage that. There's medication that we can use to manage dyskinesia. There's surgical options. There are other things that can be done about it. So it's important to kind of jot down kind of the amount of your day that you think you're bothered by it. Think about how bothersome it is and talk to your doctor about it because we can address it.
Wonderful. Thank you so much, Dr. Bega. My next question is for Dr. Simuni. Dr. Simuni, can you summarize a little bit more about what we know about the current state and any recommendations about stem cell treatments and research?
Okay, absolutely. The question that comes up frequently, I brush through that in my round the world tour of experimental therapeutics. Let's start with stem cells, right? Because it is a big umbrella term, right? Stem cell is defined by undifferentiated cell that can come either from individual themselves. It has to be regrown to become undifferentiated, right? Because fetuses have even already have differentiated to a certain degree the cells, right? They can come from another donor and have been regrown. They can come from fetal cells, and then a cell line can be developed. So there are multiple different sources of the stem cells, all of which is packaged under the stem cells, right? Now let's fast forward to Parkinson's disease. Why is there interest in stem cells, right? Because the idea is that you take quote unquote undifferentiated cell, differentiate it into dopamine producing cell, and solve the problem of lack of dopamine in the brain of people with Parkinson's disease. All very logical, seems to be straightforward, right? So where the hurdles are? The hurdles are, and those have, a number of them have been solved, a number of them have not, right? Obviously, how you deliver those stem cells, how do you make them viable so that they don't die? How do you, and again, these problems can be solved. We know that we can deliver. In majority of cases, it's surgical delivery, meaning it's surgery akin to deep brain stimulation. You don't want to throw them into the bloodstream through the injection, right? Because only tiny % of them will get where they need to be. So it's in majority of cases a surgical procedure akin to deep brain stimulation, but the difference is that you deliver the cells into the target area and basically let them survive and regrow. And we have data that we can do that. The next one, will they produce dopamine? Yes, they will produce dopamine. The next very big question, will they overproduce dopamine, right? Will they kind of go on a tour, right? I'm here, I have perfect ground, I have fertilizer, let me grow, right? And let me give more fruits than I was expected to. That is a question. And the biggest question is, what if we delivered everything what I've just said? Will just correcting the level of dopamine be sufficient to fix all the problems associated with Parkinson's disease? And that's where the biggest question is, right? Because if we deliver dopamine producing cells, they will do that. But again, Dr. Mercury very elegantly covered that it is much more than the disorder of dopamine deficiency, right? So if we improve the mobility, the speed of movements, but it doesn't do anything for cognition. It doesn't do anything for the blood pressure. And for all the other problems, are we going to improve someone's quality of care? Right? So that's the background, where the studies stand. Some of you might know that the story of stem cells started with fetal cell transplants more than 20 years ago by now. In the 90s. There was a lot of ethical controversy of using fetal cells, but the bottom line is that all the studies were negative. Cells survived, cells regrown, we saw on the scans that they've regrown, but people didn't get better. And it was put on the shelf for 20 plus years. The programs have been revived with a modern technology using now cell lines, not fetal cell lines, and with better technologies. There are a couple companies that actually are running studies. There is a program in United Kingdom that is running actually fetal cell transplant studies. So the programs are going in. They're positioned for people with more advanced disease who have clear Parkinson's manifestations, who have developed certain degree of motor fluctuations and or dyskinesia, right? So obviously ethically positioning it into people with at least moderately advanced disease. Blue Rock Therapeutics is one of the companies. We will be participating in those studies. So scientifically, extremely interesting. Patients are extremely interested in stem cells. Everyone is enchanted by stem cells. There are a lot of questions to be answered. Now, the last sentence, the word of caution. And there was a reason why I started with a big umbrella of stem cells. There are a lot of shops, and I'm purposefully calling them shops, that are advertising stem cells, autologous infusions for myriad of the diseases, including Parkinson's disease. Beware, those are not studied. Those are not regulated. For some reason, stem cell infusion therapies do not require, these kind of stem cell infusion therapies do not require FDA approval, FDA review and approval. So be very well aware of that. And I would be very clear and transparent that these kind of stem cell therapies do not have any evidence to support efficacy in Parkinson's disease. It is out-of-pocket expenses and charges. We do not encourage these kind of therapeutics.
Thank you so much, Dr. Simuni. Appreciate it so much. My next question is for Dr. Akhtar. Dr. Akhtar, will the test for the new synuclein biomarker be available for patients with Parkinson's disease? And if so, where?
It's a good question, very popular question. I think, yes, it will be. At the moment, the synuclein seeding assay test does have approval from the FDA as a breakthrough device. And that designation does not require a clinical trial. It's more of a designation where there is a new development. It is significantly different from what's currently available. It answers a specific question, and there's a chance that it will help patients and their care providers in managing their disease. And it's a revolutionary change. So all of those criteria were met. And so the FDA granted that approval back, I think, in February or March of this year. And actually had granted some approval for this same test in Alzheimer's disease a few years before. So we are looking into how to make this test available for patients. It's going to take some time. Obviously, the biggest drawback at the moment is that the test requires a cerebrospinal fluid sample. So that is not a sample that you can walk into LabCorp or Quest or even a local hospital lab and have collected for you. That has to be collected usually by a neurologist in the office. And once a sample is collected in that way, we have to ensure that it gets sent to the company that does the test and that the result comes back. And we're also working out all of the payment and the insurance approval and all of those things. Right at the moment, none of these tests are approved by insurance. They are not approved by Medicare. So I'm hopeful eventually they will be available. I have a feeling that, like the DAT scan, they will be available, but they may not be useful in every clinical scenario. And I stress this because I think that any of these tests, these new biomarker tests, really should be sent by a movement disorder specialist who's had a chance to see you, examine you, talk about the test with you, and what the pros and the cons are of the result. And it's very important versus a test that can be sent by maybe a non-specialist. I don't think we're ready for that, and I don't know that we'll ever really be ready for that. I think that this is going to be a test that's going to be used as a part of a more comprehensive diagnostic scheme. So stay tuned. This is relatively new for all of us, and so we're working on how to develop this for clinical use.
Wonderful. Thank you so much, Dr. Akhtar. My next question is for Dr. Mercury. How do I recognize signs of dementia?
That's a good question. It can be quite difficult to recognize signs of dementia because often we'll think, well, it's normal aging. I think a good rule of thumb is if your family is concerned about your memory, see your primary care doctor first, see if there's any changes they're seeing, for example, vitamin B12, we say round up the usual suspects. see your movement disorders doctor and see if there's anything from medication that they might be concerned about that could be causing you some difficulties. And if all of those have been addressed and still there is a concern that you have or the family has, I think it is a good idea to get neuropsychological testing. That creates a baseline. It looks at strengths and weaknesses. At the very least, it can be very reassuring that it's normal aging. At the same time, if there is something that's just starting to appear, the earlier we can catch a cognitive disorder, the more we can typically do about it. And it gives you the information that maybe you do need reminders or you need assistance with medication. Of course, we all know taking medication for Parkinson's can be very complicated, has to be taken frequently. And the worst thing that can happen is if someone does have a memory problem and is forgetting to take their medication and yet goes to their specialist in movement disorders and says the medication's not working because they don't remember that they're having trouble with taking it. And then the movement disorders doctor has to figure out why isn't it working, and is it because you're not taking it properly, and so on. So I would say, rely on, if you have any suspicions that maybe your memory's a little bit worse than it should be. If your family has suspicions, see your team of doctors and then consider if they recommend it, neuropsychological testing. It is covered by most insurances.
Wonderful. Thank you so much, Dr. Mercury. All right, my next question is for Dr. Bega. Dr. Bega, is there a best diet for Parkinson's disease? And can you address protein and how it relates to medication efficiency?
Right. This is a question we get a lot, understandably. If there's something you can do for yourself, you want to be able to take as much control over your disease as you can. And what we know is it's a little disappointing because it's really hard to study diets. As you can imagine, trying to control everything that people eat in order to know what is actually making an impact and what isn't is difficult to do. We do think that having a healthy diet that minimizes vascular risk factors is important. And a diet that is generally healthy for our cells in our brain would be one that has a lot of antioxidants and minimizes inflammation in our body. Now, this is all theoretical. Some of this is based on animal data. Some of this is based on specific products that have been studied, mostly in the laboratory, that show protective effects on cells. Nobody can actually say that a particular diet has been proven to be more effective than another. They've done studies, though, showing that people who generally follow healthier diets tend to live longer, of course. They tend to have less vascular problems, and less vascular problems means their brains can remain healthier longer. Again, less inflammation theoretically would mean that the cells in the brain might stay healthier longer. So an example of this would be a Mediterranean diet. Mediterranean diets have a lot of leafy, colorful vegetables and fruits, healthy fats. We have a lot of omega-3 fatty acids in them. They have a little bit less of inflammatory products in them, like simple sugars, which could be bad for the body and for the brain. Similarly, like the DASH diet, which is sort of a heart-healthy diet, can reduce vascular risk factors, and that can be good for the brain. Avoiding inflammatory foods like a lot of desserts and sugars and simple carbohydrates like white pastas and white breads and things like that, because those can lead to inflammation in the body. Caffeine is okay for people with Parkinson's. We actually have some positive correlations between coffee intake and Parkinson's disease. Alcohol, probably not as good for people with Parkinson's disease. That's not to say that you need to eliminate it, but reducing the amount that you take compared to what you may have used to had throughout your life. So those are just some general guidelines. We have some guidelines that we provide to people that we have as handouts in our clinic and that we can share. And the Parkinson Foundation also has some information on diet. But again, these are general suggestions, not hard and fast rules. Oh, and regarding protein, which you asked me about, another important question. So you need to have protein in your diet. We do know that some people are more sensitive to absorbing their levodopa when it's taken close in time to when they have protein in their stomach. And so generally we try to have people avoid taking their carbidopa levodopa with high protein foods. within about an hour, if possible. We know that that's not always possible. Some people require their carbidopa-levodopa very frequently, and that becomes really prohibitive. But the sensitivity to protein and how it affects the absorption of your carbidopa-levodopa will differ from person to person. And so we have to see kind of how it affects you and how much caution you need to really take in terms of how much space to put between your protein and your pills. And one suggestion, if you are someone who does notice that you're sensitive to that interaction, that your meds don't work as well if you take them too close to protein, is to try to shift more of that protein into the evening time, so that you get good absorption of your medications during the day and have more of your protein later on in the evening. That's one suggestion that can help.
Thank you so much, Dr. Bega. So we just have a few minutes left. My last question is for Dr. Simuni. Do you soon think that there will be drugs available to help slow the progression of Parkinson's?
I certainly hope. And again, that's where tremendous amount of research is. You saw my slides, you saw the number of drugs going into the therapeutic. So simplistically, I guess quantity should lead to quality, right? But more importantly, I believe that the current studies are designed smarter and are targeting underlying disease biology, all of which are the essential attributes for success. Targeting disease biology, targeting specific Parkinson's-related genes. I'm not saying that it's easy, and I'm not going to give you the timeline, okay? We're making progress. I've shown one of the slides. It took Alzheimer's disease 20 plus years to cross not the finish line, but the starting line. But look, 3 drugs in 1 year plus have read out positively for amyloid targeting therapeutics with all political controversies, with all. So again, what we need funding to conduct those studies. They're extremely expensive. We need scientists to dedicate their time. We need patients to participate in the studies. We need pharma to continue to be interested, and all of that is happening. So I will finish with where I started. I'm very optimistic. The field is booming as never before. It's a challenging question.
Okay, and with that, thank you, Dr. Simuni, and we will conclude the Q&A portion of today's webinar. And I want to extend a huge thank you to all of our presenters and panelists today. So in closing, I want to thank you all again for joining us today. We greatly appreciated having you here with us on your Saturday morning. We know your time is precious, so thank you for spending it with us. At Northwestern, we really pride ourselves on providing a comprehensive care to everyone seen in our clinic. In addition to having a team of healthcare professionals, our Parkinson's Disease and Movement Disorders Center offers a wide variety of support groups and classes to further support your well-being. And with every year, we continue to strive to add new and innovative and supportive programs to our existing offerings. I wanted to remind you all that a recording of today's virtual symposium, along with resources discussed, including my contact information, will be sent out to everyone in a follow-up email within the next week. Thank you again to our presenters and our panelists for sharing your time and sharing your expertise with us. And thank you again to our sponsors, Amneal, AbbVie, and Supernus, and a huge shout out to the Parkinson's Foundation for co-hosting this event with us. A final reminder to please fill out your evaluations and let us know how we did today. And again, I want to thank you all for joining us today, and we hope to see you again very soon.